Next is, what are the different cells of immune system? So understanding cells of immune system, they may not ask direct questions from cells of immune system, but if you have a clear idea what are all these cells going to do, it makes your job easy in understanding the different types of immunity, cell mediated and humoral immunity, what is their mechanism and pathogenesis. So we'll begin with, there is a specific classification for it. This is how we are going to discuss it. One is lymphocytes, and the other is antigen presenting cells, and the third one is the effector cells. When it comes to lymphocytes, as we all know, B and T lymphocytes, but after the yesterday discussion, you also know that the natural killer cells are large granular lymphocytes. They are a type of lymphocytes itself. So we'll see the B and T lymphocytes, how they work, what is the mechanism of action, how they protect our body against the infections, followed by antigen presenting cells. What are the examples for antigen presenting cells? We do have macrophages. Okay, we main important antigen presenting cells. A topic of discussion is dendritic cells, very important. We'll discuss the types of dendritic cells. You know, even B cells, they also act like antigen presenting cells, okay, because they present antigen to T helper cells. Okay, so that's the reason even B cells are a category of antigen presenting cells. So these are the three examples. So what are the effector cells then? The effector cells are the cytotoxic T lymphocytes which are released whenever there is contact with the antigen, and also the B lymphocytes which are released, okay, on contact with the antigen. These are all the effector cells. Even sometimes the neutrophils and eosinophils in response against infection, these are also effector cells. These are almost the last cells to act. So that's the reason we call, they're acting at the effector end. We call them as effector cells. So we'll be discussing the lymphocytes, APC, and effector cells in detail in the next part of the discussion. So we're starting with the lymphocytes. Lymphocytes we have, on an average, we all know that the predominant component of leukocytes in the body, when you do differential leukocytes, first is neutrophils. They make up to 60% of the body. This neutrophil is followed by lymphocyte, which accounts to 20 to 45% of the cases. Okay, 20 to 45% of the number. In total, these are not important, just for a general idea and all. This is how a lymphocyte looks. It's a small round cell with a spherical nucleus. One interesting concept is like lymphocyte recirculation. These lymphocytes, they are wandering cells. They won't stay at a single place. They go to lymphatic organs like spleen, lymph nodes, and liver. They again come back to the blood. They travel through the lymph. They again enter lymphatic organs. So there is a constant traffic of these lymphocytes traveling through the blood, lymph, and lymphatic organs. We call this as lymphocyte recirculation. So that's the reason, though the lymphocytes come under adaptive immunity, they are trying to be readily available to act against any foreign antigen. So they recirculate. And when you check the blood sample and all, the recirculating cells are mainly T cells, and B cells, they prefer to be more sessile. They stay inside the lymph nodes in the reticular, perireticular areas. They prefer to be sessile, but recirculating cells are mainly the T cells. So they're asking, how will you differentiate B cell from a T cell? There are many features, but an extra point what you'll remember is, most of the times the T cells are recirculating, whereas most of the times the B cells are sessile. And when you discuss lymphocytes, the topic is incomplete without knowing the cluster of differentiation. As we all know, these lymphocytes, as I told you yesterday, the T lymphocyte has a CD3 on its surface. The B lymphocyte, what does it have? It has immunoglobulins on its surface. And we also have, on the natural killer cells, okay, on the natural killer cells, we have CD16 and CD56. What are all these? They are a characteristic antigens specific to a particular cell. When you want to identify which kind of cell is forming tumors, which kind of cell, which kind of a lymphocyte it is. So these surface antigens or markers, they have been identified on these lymphocytes, okay, and they reflect the stage of differentiation of this lymphocyte and also the functional properties of the cell. And if for a particular lymphocyte, we have over 150 CD markers, 150. So that's the reason when we read about WBC neoplasms, the important parameter for MCQs and also theory exams was like for which, so you take example of follicular lymphoma, there are specific CD which are specific to follicular lymphoma. Similarly, Burkitt's lymphoma. That's so many lymphomas and leukemias. They are categorized or they are identified to the cell of origin is identified based on these CD markers. And I have mentioned few important cluster of differentiation markers, okay, and CD4 and CD8 and CD3 are specific to T cell. CD1 is specific to thymocytes and Langhans cells, which are dendritic cells. CD19 and surface immunoglobulin. Surface immunoglobulin is more specific for B cell. And last, CD16 and 56 are specific for natural killer cells. So they might ask, what is cluster of differentiation? Mention the CD markers are specific to the normal lymphocytes. So beyond that, the cancer CD markers and all not important for our exam. Okay, so let's quickly go to the differences between T and B lymphocytes. T lymphocytes, as I already told you, peripherally they are wandering, they roam here and there. So that's the reason 60 to 70% of the peripheral lymphocytes are actually T cell lymphocytes, whereas B cell lymphocytes are only 10 to 20% of the lymphocytes. And next important thing is T itself is they mature in the thymus. Okay, B means they mature in the bone marrow. That's the reason these are thymus derived, these are bone marrow derived lymphocytes. Major maturation process occurs in the respective organs. And what kind of effect or mechanism or response is seen whenever these cells are activated? When T lymphocytes are activated, we'll see cell mediated immunity, and also there will be helper cell production, which in turn, helper itself is the name given because it helps B lymphocytes to produce antibodies. Okay, as I already told you, B lymphocyte acts like an antigen presenting cell. Whenever there is an antigen, it takes the help of helper T cell and it will produce antibodies. So this is how the cycle runs. Okay, so T lymphocytes, they also have a role in humoral immunity, but not directly, they only help. So that's the reason we call it as helper cells, which help in antibody production. Humoral immunity is production of antibodies, and what type of antibodies are produced, how they act, the classification, the mechanism of action, we discussed yesterday. Next important thing is the T lymphocytes, they do not detect free or circulating antigens. Majorly, T lymphocytes, they focus on intracellular antigens. Okay, T lymphocyte, they focus majorly on intracellular antigens. So whenever there is an intracellular antigen, the cell itself will detect this antigen and it will project the antigen onto the surface of the cell with the help of a complex called MHC1 or MHC2, based on what type of cell it is. If it is antigen presenting cell, it has MHC2 on its surface. If it is normal healthy cell, it will have MHC1 on its surface. So whenever there is an intracellular parasite or virus or bacteria or cancer cell, anything which is intracellular, the response is by T lymphocytes, because T lymphocytes, they cannot detect free or circulating antigen inside the blood. But what is the speciality of B lymphocytes? The B lymphocytes, they recognize antigen by means of IgM class. So they actually, yes, they are also getting help from the macrophages because they don't go and carry an antigen. In the blood, the antigens are traveling there, and in the blood, the macrophages and all, they take this thing and they present it to the they present it to the immunoglobulin, that is the B cell. And on the B cell, the specialty of B cell is B cell has surface immunoglobulins, that is the marker. The IgM which is present on the B cell, it takes the antigen now. Okay, it takes the antigen, and this, after interacting with the antigen, the B cell will now release antibodies into the circulation. That is a humoral mediated immunity. So we'll see the image clearly, but my point here is, when you're writing about T lymphocytes, focus that they do not detect any free or circulating antigens. The free or circulating antigens are actually detected by the B lymphocytes. And MHC is important in presenting the antigen here. Okay, and also T lymphocyte, the main types are CD4 and CD8. CD4 is majorly for T helper cell response. T helper, we call it as a T helper cell. CD8 is mainly the cytotoxic lymphocyte which directly kills the cells, and this helps in release of antibodies, and this is direct cell lysis. Okay, this is the difference. But come back to the B lymphocyte. Whenever B cells are stimulated, they differentiate into a stage called plasma cells. So plasma cell is a differentiated B cell which actually releases a lot of immunoglobulins. You know what are the types of immunoglobulins. So in this previous part, while we are discussing the cells of immune system, you can focus the importance of MHC. What is MHC? It is major histocompatibility complex. What is the other name for MHC? It is HLA, human leukocyte antigen. And what is that chromosome which is responsible for synthesis of MHC? Chromosome 6. Chromosome 6, the cluster of genes which are present on the chromosome 6, they help in synthesis of this MHC. So basics are done. Let's not go into the details of the basics, but what is more important is differentiate MHC1 from MHC2. Their basic function is they present antigen, but they are not antigen presenting cells. They are actually a complex which is present on the antigen presenting cell. If APC, you take an example of APC, that is dendritic cell. If this is the dendritic cell which has a dendrite like structures, okay, if this is an APC, the hand for this antigen presenting cell is MHC. You consider APC as a whole body, the hand of the body which is actually giving the antigen peptide is MHC. That's it. This is how simple it is. So what is the structure of this MHC and on which bodies these hands are present? Okay, and how they are coded. Okay, how they are coded and how they act and all is our topic of discussion. So chromosome 6 codes them. Okay, and what are the loci? HLA A, B, C loci. They encode MHC1, whereas HLA-DR, HLA-DP, and HLA-DQ are the one which are specific for MHC2. These are all genetic parts. If you want to forget, you can. They are not important for exam. But more important is MHC1 is present on all nucleated body cells. Why I am stressing on all nucleated body cells? Because the nucleus, which is important in synthesis of the protein, and also the rough endoplasmic reticulum, which will actually help in the process of protein synthesis because it has ribosomes. So the transcription part, translation part, they should occur so that MHC as a protein is secreted. So what are anucleated body cells in the body? They are RBC. So the RBCs, they lack MHC1. RBCs, they lack MHC1. Platelets are also, actually platelet is not a cell. Megakaryocyte is a cell and platelet is actually a detachment of the arms. Okay, detachment of the arms. That's why when you see a platelet, you won't see any nucleus in it because it is released whenever it is ready to be released by megakaryocyte. So at the time of release itself, the nucleus of the megakaryocyte will impart. What does it do? Will impart HLA or MHC to it. So that's the reason it is located on all nucleated blood cells, body cells, and also the platelets. So RBCs, they lack it. RBCs, they lack it, and also the dead cells, which cannot are nuclear cells, they'll not have this MHC1. Where do you see MHC2? MHC2 is specific for the antigen presenting cells. The examples of antigen presenting cells, you are already aware of them. One is dendritic cell, other is monocytes or macrophages, and last is B lymphocytes. So next, the structure part, don't worry, it may not be important, but if you want to remember, I'll tell you one thing. Here, MHC class 1 and MHC class 2. Class 2 is easy because it has alpha 1, beta 1, alpha 2, beta 2 structure. And in between both alpha 1, alpha 1, beta 1, you have a peptide binding cleft. This is antigen presenting cell, and this is MHC complex, and this is the peptide binding site, right? And this is the antigenic peptide. So this cleft is formed between alpha 1 and beta 1. So the remaining are alpha 2 and beta 2. That's it. That is MHC2. When it comes to MHC1, the peptide binding cleft is in between alpha 1 and alpha 2. And what structures you'll see? Alpha 1, alpha 2, and alpha 3 are the three subunits. You have beta 2 microglobulin also here. So this is how you have to remember the structure. Only two important points you need to remember. So let's go back to the MHC again. You know where they are located now. You know how they are encoded, and you know their structure in detail, in crisp way. Okay. Next, how they function. How they function. You take MHC1 or major histocompatibility. The name itself says it is a peptide display system of adaptive immunity. So what are the two types of adaptive immunity? One is humoral mediated immunity, and other is cell mediated immunity. Okay, one is humoral and other cell mediated. So the MHC1, what it is doing is, MHC1 is continuously present on, like what does it do? It is present on all the living cells, all the human cells with nucleus. So it is constantly and continuously sending a peptide signal with the help of MHC1 to the T cells, okay, to the T cells. How, like why it is happening on a continuous basis? Because we don't know when we have a viral infection or a cancer or any dysregulation and all. So this is a continuous process, continuously presents endogenous fragments of proteins located in the cells. So this helps rapid detection and destruction of infections with intracellular pathogens. So I'm only referring to the cell mediated immunity here. Okay, so and then whenever the MHC1 is presented, whenever the MHC1 presents an antigen, the T cell comes and binds to it with the help of T cell receptor. Okay, and this will activate which system in the body? It will activate the inflammation and release of cytokines, which leads to death of the cell, or this activates and kills the whole of the cell with the release of perforins, and we call it as a cytotoxic T lymphocyte. We'll discuss in detail in cell mediated, don't worry. Let's go back to how MHC2 functions. MHC2 function is antigen presenting cell. And what does it do? It is antigen presenting cells like B cells, dendritic cells, and also the macrophages. Macrophages, they are present in the blood system. Dendritic cells more or less beneath the epithelial barriers. So what do? They take extracellular peptide, okay, or humoral or blood circulation. The peptides or antigens which are traveling in the blood circulation. All these are phagocytosed. Okay, so whenever they look at an antigen, the antigen presenting cell will engulf it with the help of endocytosis. So the antigen now enters inside. The antigen is broken into pieces. The peptide is now presented on with the help of MHC2. Okay, so they present it to CD4 T lymphocytes, CD4 T lymphocytes, which are helper cells. So whenever they present to T cell receptor of the CD4 lymphocyte, now you won't see the cytotoxic response here. Instead, you will see activation of the B cells by CD4 T lymphocytes. So what happens whenever B cells are activated? They turn into the plasma cells, and plasma cells will release antibodies, and in turn MHC2 is actually helping in an indirect way. Though it is acting on T cells, in an indirect way it is helping humoral mediated immunity, whereas MHC1 is helping cell mediated immunity. Okay, so these are the two important things from MHC. And there is MHC3, just for the topic of completion I'm discussing it. MHC3, the examples for MHC3 are also, we call it as class 3 molecules, are complement C2, complement C3, complement BF, and also tumor necrosis factor and lymphotaxin. This is a common question in PG entrance examinations. What is MHC3 and what are the components and all? But for us, what you need to remember is, your take home is, MHC3 does not take part in the peptide display. So it will not help in peptide display. Rather, it has some complement and also the cytokine production. Both complement components and also the cytokines are produced by whatever MHC3 gene which is present on the chromosome 6. So this will conclude our indirect discussion on MHC. But let me add, because sometimes the examiner is fond of asking definitions and all. So what is MHC restriction? The problem, like the research into MHC and our HLA, and why do we do HLA typing for before performing what we call it as the transplantation. Whenever we are going to perform a transplantation, we do HLA typing or matching between donor and recipient. And why do we do that? Because in the initial stages of this transplantation era, in the era of transplantation, there used to be so many graft versus host disease and graft rejection cases. So when they went into study, they have found out that there are these specific MHC components, histocompatibility complexes, which are present on the cell surface. Like all the living cell surfaces are antigen presenting cells present in the tissues. And what is the problem with these MHCs? The problem with these MHCs is they are present on the cell. Whenever they are present on the cell, they are not antigens. Actually, they are helping in presenting antigen to our body. But when they enter a different cell, these MHC arms, they now act like antigens, and they inflict or the damage. Okay, they inflict damage and they cause graft rejection in the patients. Okay, so our body, whenever we are transplanting a liver, and if there is no proper HLA typing, our body will recognize all these HLA from other person like to be antigens, and it will start mounting antibody response, damaging the liver, and there will be graft failure. So this is what happens. So what is MHC restriction then? In each person, the T cells of our body recognize only peptides which are displaced by that person's MHC molecules. Okay, if there is a person X, and if this is the T cell of person X, it will only detect the peptide presented by the MHC molecule of person X. It will not detect the MHC molecule peptide, the peptide present by the MHC molecule of person Y. So this is MHC restriction. Okay, it's a simple thing. And that's the reason whenever the person Y comes, instead of detecting the peptide, the T cells will detect the whole of the MHC arm as a foreign antigen, and they'll mount a immune response. So what we're doing, let me go back and make sure that we are going on track. See, you know the innate and adaptive immunity. You know what are the cells of immune system. You know clearly the lymphocyte, what is CD, and what is the basic difference between T and B lymphocytes, and the difference between MHC1 and MHC2 with all the classic stuff, and also the image of the MHC and its structure, and what is MHC restriction. So T lymphocytes, while we are discussing, we have come across two stuff. One is CD4 positive cells. We also call it as helper cells, and other is CD8 or cytotoxic cells. I need not explain you all these things now. See, 50 to 60% of the mature cells are CD4, and like this is not. I don't think it is 50 to 60. it should be less. Okay, so it will be around like the remaining all mature T cells are actually the cytotoxic T cells. And the CD4, it binds to the class 2 MHC. CD8 binds to the class 1 MHC. This is how I remembered. Like if there is a 2 into 4, it is equal to 8. If there is 1 into 8 is equal to 8. So I remembered 8, and if I get a CD4, then it is MHC2. If I get a CD8, it is MHC1. Okay. Next is helper cells and cytotoxic T lymphocytes. Helper cells, they help in secretion of antibody by B cells. Cytotoxic T cells, they directly go and they kill the virus infected or tumor cell. These are the differences between CD4 and CD8. What is T helper response? There are three kinds of T helper response. One is T helper 1 response, T helper 2 response, and T helper 17 response, which is not important. But T helper 1 and T helper 2, in the helper cells itself, based on the cytokines which are released. If the cytokine which is released is interleukin 2 and interferon gamma, the response will help in combating the intracellular parasites. Okay, but if the helper response, if the cytokines which are released are 4, 5, 6, 10, and 13, then the response is predominantly on the extracellular parasites or elements. So only remember this much, not going into the details because they might not ask this, but the cells of immune system is in the topic. And when there is a T cell response, it can be a T helper or cytotoxic. And in the T helper, we have two, T helper 1 and T helper 2. They are differentiated based on the cytokines released. You know what are all the cytokines, and the response is different. T helper 1 has intracellular response, T helper 2 has extracellular response. That's it. So the other types of T cells, I'm not going into much details. What we are missing here is the natural killer cells. Yesterday we discussed in detail about the natural killer cell, the count, the look, and how do they function and all. But here there is a specific entity or a special entity called NKT cells. It has both the components. One is natural killer cell and other is T cell. You might have remembered the natural killer cell will have only CD16 and 56, nothing else. But if there is a cell with both CD16 and CD56 positive, along with that, even if the CD3 immunoglobulin IgM or something else like which is specific to, sorry, not IgM, CD3, CD4 or CD8, which are specific to the T lymphocytes. If they are also present, we call it as NKT cell, both natural killer and the T lymphocytic cell. What is its function? These NKT cells, they also recognize microbial glycolipids like T cells, but whatever they do, their function is not drivers. They only can detect limited antigens, but T cells have the antigenic diversity. They can recognize almost all the foreign antigens, whereas the NKT cells have they have less diverse receptors for identifying. Okay, so that's the basic difference. So I'm not touching regulatory T cells. When I say regulatory T cells, yesterday we discussed they are a part of uh, what we call it as a peripheral tolerance. Okay, and whenever there is a dysregulation of these regulatory T cells, it leads to autoimmunity. And what do? They control. They make us tolerant towards our own self antigens, right? So that is the function of regulatory T cells. So this is all about antigen presenting cells. Antigen presenting cells, I already told you that dendritic cells, macrophages, and B cells. Dendritic cells are three types in general. One is interdigitate dendritic cells, other is plasmacytoid dendritic cells, and third variety is follicular dendritic cells. What is the difference between these three, and why it is important you to know about these things? You know the Langhans cells. Langhans cells are a part of dendritic cells, and what do? They are strategically placed under the epithelia of the body to capture and present antigens to T cells. So these are antigen presenting cells, and they are present below the epithelial layer. Okay, with the dendritic process. They have dendritic process. Okay, and what do? Whenever there is a foreign antigen, they'll quickly recognize it. We call them as Langhans cells, and they present it to the T antigen. So this is the interdigitate. So this is how they digitate in between the cells, which I had just done. Okay, they interdigitate in the epithelia and they recognize it. And next is plasmacytoid disease. Okay, plasma, they are inside the plasma like cells. Normally the disease have digitations, but they stay uh, yeah, the main function of natural killer T cells, as I already told you, they function similar to the T cells, like what we have seen here. I'm sorry, I'm going back. So what you have seen here, they play a role in defense against some of the infections. What some means, like almost all, but the problem is they are not diverse, or they won't have much action similar to the T cells. So you have to ignore and neglect them. They might be a hybrid form of both natural and killer and T cells. Okay, so their role is prevention against some of the infections. Not all, because they're not diverse. And the plasmacytoid disease, they are present in the blood and lymphoid organs. Okay, and whenever they come across an antigen in the blood, they engulf it, okay, with the form of endocytosis, and they help in releasing one more important function of plasmacytoid disease is they also have potential to release interferon type 1. Okay, and interferons, hope you might have seen the video. Interferons are actually agents which are produced by our body to fight against viruses. Okay, so this plasmacytoid disease might produce this antiviral cytokine. What is that? Interferon type 1. And next is follicular dendritic cells. They have a follicular like structure. They are located inside the germinal centers of the lymphoid follicles like this. Okay, in the spleen and lymph node. And what do they do? Whenever there is an antigen, okay, if this is an antigen and if this is an antibody complex, whenever there is a primary immune response, this antigen is now presented to the B cell, which is present inside the spleen and lymph nodes. And what do? They give rise to secondary antibody response by trapping the antigens which are bound to antibodies and complement. Okay, and presenting them to activated B lymphocytes in lymphoid follicles. So in the circulation we have antigen antibody, and what do? They trap this inside the structure, that is the lymphoid follicles. After trapping the component, whenever there is an antigen, whenever there is an antibody, there is its complement, its friend, it is always there. And whenever this reaction is going on, already the antibody is trying to fight against this antigen. If this primary immune response is not sufficient, this complex is taken, engulfed by the follicular dendritic cell, and after engulfing, this follicular dendritic cell will now present this complex to the B lymphocytes, the activated B lymphocytes which are present in the lymphoid follicles, so that the activated B lymphocytes now they form plasma cells, and the plasma cells will now release more and more antibodies to fight against this antigen. So what they're doing, they're producing a secondary antibody response here. So this is follicular dendritic cells, and we're done with interdigitate and plasmacytoid disease, and we're discussing what are all the different cells of the immune system. And macrophages, they ingest microbes and they present antigenic peptides to T lymphocytes. Okay, so they help in more or less the cell mediated immunity. Okay, and B cells, what do? They present antigens to T helper cells, as I already discussed 3 to 4 times, so I'm not stressing on it. Just read it in the notes. So there are few more effector cells at the end point. These are the natural killer cells, which are also part of immune system. And what is the function of natural killer cell? Yesterday I told you one point, right? So natural killer cells, they have both inhibitory and stimulatory response, and it has relation to the MHC1 expression on the body, right? So whenever there is a MHC1 expression, the inhibition takes place. Natural killer cells won't act, but whenever these you need to understand this. This is important. This is a healthy cell. Healthy cell has MHC1 complex, and what is the function of MHC1? Whenever there is a peptide or an antigenic or a viral infection in the body or a cancer in the body, there will be some antigens which are produced inside the cell. The MHC1 will immediately process this antigen and it will place a peptide on its surface so that the T lymphocytes can detect it. Okay, T lymphocytes, as I already told you, they are wandering. Okay, they go here and there. And what do the T lymphocytes do? The T lymphocytes have the T cell receptor on its surface. So this T cell receptor on T lymphocyte will identify it and it will produce a response. This is helped by MHC1 and immediately the cells are killed. But problem here is the virus, it will evolve itself to adapt to this condition. So what does most of the viruses do? They downregulate the expression of MHC on the cell surface. So the virus, for it to survive, it downregulates the MHC1. It won't allow MHC1 to reach the surface of the cell and present the antigen. So slowly what happens in virus infected cells, they are stressed cells. They become, there will be less number of MHC class 1 expression on the cells. So whenever there is less number, the inhibitory is gone. Now the NK cells will get activated. Now the NK cells will act on these virus infected cells. So you need to understand this difference. Well and good, MSC1 is doing fine, then the T cell has more antiviral property. But if MSC1 is downregulated, now the NK cells are natural killer cells, they react against these stressed cells. Okay, so this is the mechanism behind it. And we also have antibody secreting plasma cells and also macrophages. They're all effector cells of humoral immunity. And CD4, CD8 T cells, they are effector cells of cell-mediated immunity. And even neutrophils and eosinophils also help in. We also call it as a natural immunity or native immunity. Okay, it is natural, it is native, and it is readily available whenever there is an infection. So the immediate action is there which can give response to infection. Okay, for example, you take the epithelial barriers of skin, GIT, respiratory tract, and also the phagocytic leukocytes, natural killer cells, and complement system. These four are actually the examples for the innate immunity. And when it comes to acquired immunity, we also call it as acquired or specific immunity. This acquired immunity part, it is more specific and powerful, but it is not readily available. You need a prior antigenic stimulus so that the acquired immunity can act. Okay, so there are two types of adaptive immune responses whenever you come in contact with an antigen. That is the humoral immunity, which is mediated by the antibodies produced by B lymphocytes, and also the cell mediated or cellular immunity, which is mediated by the T lymphocytes, also called as T cells. So these are the two classic examples for acquired immunity. We will go into the next part. Innate and acquired is done. This is simple. They'll ask you for 2 marks, differentiate innate and adaptive. You know the examples for each, and you know the basic difference between each and the different names for innate and acquired immunity. So if you observe here, sorry, so innate immunity, whenever there is a microbe at the epithelial barrier, you have phagocytes ready to eat the microbe whenever it enters. So whenever the microbe is eaten or taken away by the phagocyte, it is readily presented to the complement system of the body. Okay, and also the natural killer cells which are readily available. So while we're discussing the natural killer cells, I did tell you a point. What was that? In the natural killer cells part, I did tell you it is a part of innate immunity because it doesn't require any prior stimulus. It directly acts on the microbes. So that's the reason we categorize into innate immunity. But the same natural killer cell do have an adaptive immune system also. That is what is antibody dependent cell mediated cytotoxicity. Let's quickly move to the adaptive immunity part. In adaptive immunity, you won't see immediate response. The microbe is now, the antigen is now presented to either B lymphocytes or T lymphocytes. Okay, and then the adaptive immunity starts and it will produce antibodies. in case of B lymphocytes, or it will produce effector T cells, that is cytotoxic T cells in case of T lymphocytes, and all these at later part will fight against the infection. So this is the basic difference. This is the dendritic cell with antigen. Okay, it has a dendritic processes, you can see, and it has an antigen and it has MHC2 two component MHC2, and you can clearly see the antigenic peptide here. It is taken into the splenic sinusoids or the lymphoid organs and all. Okay, this is a lymph node. I'm sorry, this is not splenic. It's a lymph node. So after entry into the lymph node is rich in both T and B lymphocytes, right? It is rich in both T and B lymphocytes, and you can see here for humoral mediated immunity, for the response to occur, we got two things here. One is CD4 and other is CD8. In CD4 T cells, we also see whenever there is an interaction between an antigen presenting cell and CD4 T cell, there will be a T cell proliferation and a differentiation. So this is the step which occurs, and it occurs with the help of release of cytokine, mainly interleukin 2. So based on which type of interleukin is released, if it is 2, you will see T helper 1 response. If it is something else, 4, 6, 8 and all, you will see T helper 2 response. Forget everything, but there will be some release of interleukin and you will see T cell proliferation. So whenever proliferation occurs, there are two kinds of cells released. One is effector T cell, which actually has an effect, and one is memory T cell, which is stored so that at the later point of time, whenever there is a superimposed or a sudden infection, the memory T cells are ready to act. So these cells, differentiated cells, be it be a CD4 effector cell or be it be, what is this, CD8 T cell. CD8 T cell, whenever they are entering into the blood circulation, they cross the barrier and they go to the site of infection or inflammation. There are two classic examples. If CD4 effector T cell is there, it again has an interaction with the ingested microbes at the site of infection, and there will be a release of cytokines. Okay, cytokines will now elicit inflammation because they have chemoattractants. They attract inflammatory cells like neutrophils. The neutrophils are policemen of the body. They act and they kill the infection there. Similarly, they also activate macrophages there. The macrophages are phagocyting cells. They eat and they kill the injected microbes. So this is how the response occurs in cell-mediated immunity. But this pattern of CD4, this pattern of cell-mediated immunity depends on the cytokine secretion. There is another pattern wherein you will see CD8 cell, we call it as a cytotoxic T lymphocyte. This is going to the site of infection, but here the infection is not extracellular, it is intracellular. The microbe is present in the inside the normal cell. So the normal cell has MHC1. So this is interacting with the normal cell, okay? And after interaction, it is releasing perforin granules. It is causing lysis of the cell. So it is directly killing the cell. So these two mechanisms are cell-mediated immunity. If you look at humoral, you will understand the difference. What is happening in the humoral mediated immunity? This is a normal B cell, and the B cell will have IgM on the body. Okay, there is an antibody already present on the B cell, and it is taking the microbe. It is taking circulating microbe. It doesn't require any antigen presenting cell per se. The microbe is taken by this B cell. Okay, and there are some helper T cells which help in augmenting the response, and helper T cells, they help in proliferation of all these B cells. So whenever there is a proliferation, the B cells, which are a bunch now, they can have differentiation into different aspects. One thing is they can become plasma cells. They release more and more immunoglobulins. These more and more immunoglobulins, they'll produce antigen antibody reaction with the help of complement system, and there will be neutralization of the microbes. So this is what happening in one spectrum. And along with that, there are some B cells which undergo class switching. What is class switching? From one type of immunoglobulin, there is conversion to another type of immunoglobulin, because IgG, there will be some release of IgG which occurs, okay, and IgM is a recent antibody which has a lifespan which is a bit little less, but IgG remains forever. Okay, IgG remains forever. So there will be some class switching which is occurring here. Similarly, they also have affinity maturation. That is, most of these cells, they also get converted into memory B cells. You know the memory T cells, so that at a later part of time, if there is any infection, the T cells are ready. Similarly, there will be memory B cells which are ready to release high affinity antibodies whenever needed. So all these things like the production of IgG or IgM, okay, what do? They act against the antigen in different styles. One is direct neutralization of the microbes, as we discussed. Other is this antigen antibody complex with the help of the FC receptor detected by phagocyte. There will be opsonization and phagocytosis by macrophages. One more thing we already discussed, FC component of the antibody is detected by the NK cell. So even now NK cells, they kill these infected cells. So this is known as antibody dependent cytotoxicity. Similarly, there will be complement system activation. So the complements will now, they cause lysis of the microbes, So all these are the things which occur. These are antibody effector functions. In the body, we have lymphocytes. Lymphocytes, they make around 20 to 45% of the body WBC in the differential leukocyte count. And there are three varieties of lymphocytes. Most of them we only know that B lymphocyte and T lymphocyte. But natural killer cell is also a type of lymphocyte. So 10 to 15% of the peripheral lymphocytes in the body, they are of natural killer cell variety. What is the difference? You take T cell. T cell has T cell receptor on the body. You take B cell. B cell has an immunoglobulin receptor or cell surface immunoglobulin. B cell function is secretion of the immunoglobulins by itself getting converted into the plasma cell. So these two, if they are absent on a cell which is looking like a lymphocyte, but TCR is absent, IgE is absent, and it is a slightly big in size, and if it has azerophilic granules in the body, then we call it as a large granular lymphocyte. So what is the other name for large granular lymphocyte? It is natural killer cell. So it contains abundant azerophilic granules. It is larger in size and it lacks both T cell receptor and cell surface immunoglobulin. And it consists of 10 to 15% of the peripheral lymphocytes. It is a part of innate immune system because natural killer cells, they do not require any any help. They straight away they act. So what do? Whenever there is an infection, be it be a viral or any stress related infection, so in stress, what happens? There is reduction of class 1 MHC molecules. As I already told you, all the living cells, they have class 1, okay, class 1 MHC molecule on their cell surfaces. There will be in conditions like viral infections and in stressful conditions, the class 1 MHC molecule content or expression decreases on the living cells. It decreases on the living cells. And how does it affect the natural killer cell function? Let's see in detail. Generally, the natural killer cells, if this is the natural killer cell, it has two types of receptors, type 1 and type 2. Type 1 is inhibitory and type 2 is activating receptor. This inhibitory receptor is the dominant one. They are more in number on the cell. And what do? This inhibitory receptors, they recognize the class 1 MHC which is present in all the living cells. Just focus here, this is very important. The natural killer cells have two kinds of receptors. One is inhibitory receptor and the other is activating receptor. Inhibitory receptors are dominant in number, and what do? They'll recognize the MHC1 which is present on the cells, and whenever they recognize all healthy cells, Okay, they down regulate the activating receptors. They'll not allow activating receptors to function, because if the activating receptors are upregulated, natural killer cell, it naturally kills all the cells. If it is activated, means it goes and kills the cell. What is happening in stressful situations? There is decrease in MHC molecule expression. So the dominance of inhibitory receptors is lost, because the dominance comes from mutual interaction. Whenever they both interact, because there is more number of MHC cells, there is release of those soluble mediators in the body, which will prevent attack. But when this is gone, let's say this is the cell, a healthy cell, it has MHC1. So because it has a MHC1, natural killer cell is coming, but here inhibitory part of receptor is active, so this is not getting killed. Let's go to the next scenario. This is a virus infected cell. Because it is a virus infected cell, there is no expression of MHC1, and now what is the one which is getting interacted is activating receptor. So now the natural killer cell is activated. The activation of NK cell leads to killing and elimination. So this is how it's an innate immune system. And throughout the discussion, have I mentioned about any antibody which is coming in picture? No. Antibodies, that doesn't play any role in natural mechanism or natural action of these killer cells. But there is another property for NK cells also. So the major function of NK cells is it's part of innate immune system. It directly detects whenever there is a virus infected cell and it kills it. So more or less, it is more important for viral infection, because most of the viral infections are intracellular. But the same NK cell, it has ability to detect IgG component on the target cells. Let us say if there is a cell on which there is an IgG fraction, the FC fraction of IgG is there, the natural killer cell can detect this. So whenever there is an antigen antibody binding, and if there is a FC component, the natural killer cell can detect it, and it can do the same function also in an adaptive manner, okay, in an acquired manner. So this function is antigen dependent cell mediated cytotoxicity. So the ADCC is expressed here. When they ask about ADCC, ADCC is similar to, it's a kind of it has a similarity to type 2 hypersensitivity, okay, but it's not type 2 hypersensitivity, because ADCC is a natural protective phenomenon. When I call it as type 2 hypersensitivity, I am referring to a dangerous or a damaging one, but ADCC doesn't damage it. It protects it. So natural killer cells, they protect the body in two ways. One, by an innate immune system by directly damaging it. Two, by antibody dependent cell mediated cytotoxicity. And these antibodies, they bind antigens on the surface of the target cells. So whenever there is an antigen antibody binding, the FC portion of the antibody can be detected by the NK cell, that too the CD16 FC receptors which are present in the NK cells. In the previous slide, I forgot to mention the CD3 is specific for T cells. The surface immunoglobulin is specific for B cell, whereas CD16 and CD56, that is the cluster differentiation markers, these are specific to NK cells. And among the CD16 and 56, it is CD16 which is taking part in this antibody dependent cell mediated cytotoxicity. So there will be a cross linking of CD16, and this triggers degranulation of the azerofrenic granules which are present in the cytoplasm of this natural killer cell. And the tumor cells, there is the death of the tumor cells or virus infected cells. So natural killer cells, along with the virus infected cell, they also kill cells which are under stress, that is the cancer cells. It has a role in the death of the tumor cells also. So you can use colors. Yes, you can use colors, pencils and sketches. You need to carry them to put it in a colorful way to impress the examiner. And CD3, don't remember this cluster differentiation. They're important for postgraduate entrance, not for you. I'm only telling what are important. CD3 is specific for T cell. CD16 and 56 are specific to the natural killer cells. Surface immunoglobulin detection. Surface immunoglobulin detection is specific to B cell. Okay, just remember these three important things. So first thing is the classification of immunoglobulins. Okay, so the whole discussion of immunology, it's more of a theory type. So I'll try to make it interesting by adding some clinical examples in between. Okay, but everything is factual. Immunoglobulin are the antibodies which are produced in the body, are a part of humoral mediated immunity or humoral immunity. Okay, because it's a soluble factor which is secreted by B cell after transforming itself into the plasma cell. So the plasma cell will release all these immunoglobulins. So how many immunoglobulins are there? There are 5 important immunoglobulins in the body. Okay, so they are D-game, IgD, IgG, IgA, IgM, and IgE. These are all simple facts. Let's not waste so much time in it. But more important thing is when they ask immunoglobulins, they'll ask you to define it. Though you have a clear idea about what is an immunoglobulin, but for you to write a definition and all, you need to be thorough with it. So immunoglobulins are glycoprotein molecules. Yes, they are proteins along with the glucose moiety formed by the immune system in response to antigenic stimulation. Okay, so antigenic stimulus is required for antibody production. And where do you see these immunoglobulins? They are found in blood, and also along with the blood, most of us know that you see immunoglobulins in blood, but along with that, it is seen in bodily secretions, and also right, IgA is seen on mucosal surfaces. Bodily secretions, mucosal surface, and blood. And what does it do? It binds to specific antigen which is responsible for its production, and at the end of the day, it will inactivate that antigen. Okay, so let me tell you, if they ask define immunoglobulin, you have to say it's a glycoprotein molecule formed by immune system in response to the antigenic stimulus. And what does it do? It is secreted in both the bodily secretions, blood, and mucosal surfaces. It binds to this specific antigen and it inactivates this antigen. So this is the definition of immunoglobulin. Next is the antibody structure. So we're discussing immunoglobulin and antibody. This is how it is. One is Fab end. Also we call it as fork-like end. It has a fork-like opening here. It is an antigen binding site. Okay, Fab end is an antigen binding site. And next is FC region. FC means crystallizable. Okay, C means it's constant and it's a crystallizable region. Okay, and it is formed by the heavy chains, only the heavy chains, whereas Fab is formed by both heavy and light chains, and they are linked together by the disulfide bridges. You can see both the light chain and heavy chains, and both the heavy chains, they are linked together, and strength is because of the disulfide bridges. And if you want to go into the depth, like what does they exactly do, we'll be discussing during the mechanism of action in detail. But what you need to remember is, when I say D-game, the different types or classification of immunoglobulins, it is not based on the light chains. It is actually based on the heavy chains. So the different immunoglobulins, they are classified according to their heavy chain character. And this FC region, it determines the isotope or isotype, and it binds to various immunological cells such as macrophages, whereas Fab region, it recognizes and binds to the antigens. And what does it do? It doesn't. There are two kinds of antibodies here. One is determination of isotype and other is idiotypes. So I'll teach you about this in mechanism, but what you need to remember is the difference between FC and FAB. Only heavy chains, constant region or crystallizable region, and one more important thing is it binds either macrophages or the complement systems. Whereas, and also it determines the isotope. Next is FAB, it determines the idiotype. It has both heavy chains and light chains. It is an antigen binding site, and it is made up of both heavy and light chains. So let's go back to different characteristics along with the examples. So immunoglobulins, they're asking the classification part. So whenever they ask about the classification of immunoglobulins, so we'll start with M because it is the largest antibody. It is located on the surface of the B cells and it has a pentameric structure with J chain. And whenever you see IgM, it indicates recent infection. So when you read about the serological diagnosis of hepatitis and HIV, whenever you see like IgM kind of thing, it indicates recent infection. Whereas IgG is not a pentamer, it's a monomer, but specialty of IgG is it is the most abundant immunoglobulin. Okay, and when do you see IgG? IgG is formed later in the course of infection. It's not early, it's a delayed formation, and it plays an important role in ensuring the long-term immunity in the patient. Okay, so these are the few important things. Rest of all you can read from the notes, but more important thing is the activation of the complement system is mainly because of IgM and IgG. So both the IgM and IgG, they activate complement system, and these two are involved in the type 2 and type 3 reactions, mainly IgG. So hypersensitivity reactions are also because of these immunoglobulins. So let's go back to IgA. IgA sometimes exists in dimeric form. So there was a question in MCQs like immunoglobulin with a dimeric structure it's IgA. It is monomeric in circulation, but whenever it is secreted, as I already told you, the mucosal surfaces are embedded with this immunoglobulin. So they are in dimer form whenever they are secreted. And what is their important? It's they bind to the pathogens on the mucosal surfaces and they act as a mucosal immunity. So whenever you think of IgA, think of mucosal immunity. That is the catch or keyword. Next is IgE. IgE is a monomer and IgD is a monomer. The good thing is IgD, it's located on the surface of mature vessels and its function is incompletely understood. So let's not burden ourselves by discussing about IgD. But IgE, when you look at IgE, the first thing that should come into your mind is atopy. Okay, so the type 1 hypersensitivity reaction is mediated by these antibodies. And what does it do exactly? So its important function is defense against parasites. So whenever it is secreted, it binds to the mast cells, it causes cross-linking in, and it releases the histamine from the mast cells, okay, and the basophils. It helps in release of histamine. This is the main function of IgE. Let's quickly look at the clinical relevance of all these immunoglobulins. IgM, so whatever blood group antibodies which are produced in the body, they are of IgM type, and it produces early reaction as I already discussed you. You have hepatitis C or IgM. If you see anti-HBC, it's an IgM. Most of the early infection is because of IgM. Just remember that point. IgG, not blood group, rhesus antibodies are of IgG type. Yes, it produces immune response, but it's a late reaction, and it indicates only the past infection, not active infection. And next is it mediates both type 2 and type 3 hypersensitivity. Okay, both type 2 and type 3. It is not allergy. It mediates type 2 and type 3 hypersensitivities. IgA, as we already discussed, it's a mucosal immunoglobulin, but more important thing with IgA is the type of IgA which is produced in celiac disease. Celiac disease is because of production of autoantibodies, and these autoantibodies most of the times are IgG, IgM type. But in celiac disease, also known as gluten sensitive enteropathy, as we already discussed, there is production of antibodies against tissue transglutaminase. So the structural relation between the gluten and the tissue transglutaminase is the reason for molecular mimicry and production of this autoimmune disease. And the antibodies which are produced against this tissue transglutaminase are of IgA variety. If you want to add a clinical relevance, this is the clinical relevance for it. Okay. And next is IgE, as I already told you, it is in allergy, type 1 hypersensitivity. Okay, anaphylaxis and allergy are example for type 1 hypersensitivity. So we'll be discussing the types of hypersensitivity reactions in detail as we go on. So next is mechanism of action of immunoglobulins. So what do we do here in the mechanism of action? So we are discussing how the immunoglobulins in the body, they are acting. Just for general sake, what do they do? An immunoglobulin, okay, or an antibody binds to antigen, okay, it forms an antigen antibody complex. Whenever there is formation of antigen antibody complex, same, there is a formation of antigen antibody complex, there are two things that are occurring. One thing is an activation of the complement pathway. Whenever complement pathway is activated, the main factor is C3A, it leads to formation of the membrane attack complex. Okay, C5 to C9 is the membrane attack complex. And what is the function of this MAC? MAC, it produces pores inside the cell membrane and in turn leading to the lysis of the microorganism. So this is the mechanism of action of immunoglobulin in a nutshell. But there are other mechanisms too. After formation of antigen antibody complex, sometimes instead of the complement pathway activation, we have the FC portion, okay, FC portion of, this is the FC portion of neck region, constant region. The FC portion of the immunoglobulins, it binds to the FC receptors which are present on the neutrophils, lymphocytes, and macrophages. And what do they do? Neutrophils, macrophages, or lymphocytes, they phagocytize. See, the neutrophils are the policemen of the body. Whenever they look at an antigen, and if this is an antibody, the complement helps in lysis. But complement, along with the complement, for opsonization part, the phagocytosis part is taken over by the FC portion. So this is one method. There is another method. Whenever there is an entry of antigen and production of antibodies, whenever there is an antigen antibody complex formation, there is another method called antigen dependent cell mediated cytotoxicity. So even NK cells, not this is not phagocytosis. NK cell will actually kill the whole of the cell which is having this antigen antibody complex. So we'll discuss ADCC in detail in the next section. But what we did just now is, you know the classification and structure of immunoglobulin in detail. You know what are the different types of immunoglobulins, their structure, their characters and examples. And you know the mechanism of action, what are the three mechanisms by which it acts. One is the lysis, other is the phagocytosis, and other is antibody dependent cell mediated cytotoxicity. Complement pathway can also be a potential question. So I'm covering the complement pathway in a nutshell so that you might expect complement pathway and its deficiencies in the next exam. Okay, so what are the different complement pathways? I'm going fast here. There are three pathways. One is alternative pathway, second is lectin pathway, and three is classical pathway. So what are all the three pathways? Alternate pathway, lectin pathway, and classical pathway. But the final destination is again C3. So if you remember C3, which is a common destination for all. So classical pathway, it requires activation of or formation of antigen antibody. Okay, so what is the basic difference between an innate immunity and an acquired immunity? An innate immunity is inherent. It doesn't require contact with antigen for production. But acquired immunity is, it requires contact with an antigen and formation of the antigen antibody complex and all. You observe alternate pathway and classical pathway. Whenever there is an infection with any antigen, that is, you take bacteria, and bacteria has a capsule around it. Okay, it has an antigenic surface around it. The body should produce antibodies. Then this antigen antibody complex, the complement pathway binds to this complex, and there will be reactions. So C1 plus C2 plus C2, C4 plus C2, and this leads to activation of the C3. Then there is production of anaphylatoxins, and C3b helps in formation of membrane attack complex, and this leads to cytolysis. Don't remember all these steps, but what you need to remember is this is the classical pathway. It comes late. It is more of an acquired kind of immunity because it requires antigen antibody reaction. Then only the complement goes and bites. But before that, there is one element which is acting first. The pathogenic surface itself creates a local environment which helps in activation of the complement. So this doesn't require production of antibodies by the body. It itself, whenever antigen occurs, it directly goes and acts. So I'll call it as more of innate immunity. I'll call classical pathway more as acquired immunity. Okay. So these are the difference between a classical complement pathway and an alternate pathway. Lectin pathway is not important. It is almost similar to the alternate pathway, but here you need the mannose binding lectin. Okay. So mannose binding lectin is an essential component or an extra component which is needed. Here you need only antigen. Here you need both antigen plus antibody, which is produced by the humoral immunity. But here you need antigen plus mannose binding lectin. This is neither a direct reaction nor it is an antibody. It is present in the body. So it also comes to a category of innate immunity, but it should be there in the body so that the reaction can occur. But what is the final result? As I told you at the end of the day, it leads to complement activation. So whenever complement gets activated, you will see the recruitment of inflammatory cells, which we call as chemotaxis. Similarly, we'll see opsonization and killing by the phagocytosis, that is membrane attack complex, and perforation and all. All these things, they occur by complement pathway activation. So your take home when they ask about complement pathway, it complements both antigen antibody reactions. That is classical. But there is another pathway called alternative, in which you don't need any antigen. And what is the central element? It is always C3, which is the central element for all three different pathways. What is the specialty of lectin? Lectin requires mannose binding lectin so that it binds to pathogen surface and then it stimulates or activates complement pathway. These are all the things which you need to know. And one more thing, just writing this element will give you more marks. So the element which is helping in chemotaxis is C5, 6 and 7 complex. The complement element which is helping in histamine release, that is the degranulation of the mast cell, which has a role in type 1 hypersensitivity, is complement 3A and complement 5A. These are anaphylatoxins. But the complement which is helping in opsonization, that is coating of the bacteria for phagocytosis, is C3B. Cytolysis or membrane attack complexes C5 to 9. And the complement with kinin, don't remember this, these are not important. Only go with 4. One is chemotaxis, other is mast cell degranulation, next is opsonization, and fourth is membrane attack complex. Try to remember these aspects of the complement pathway. So we'll finish up the complement part by discussing the discussions. Just focus on this table. There are around the 12 different complement deficiencies which leads to diseases in the body. This is a conclusive list, but you need not remember all these things. For you, more important, for exam point of view, they might ask you, mention some of the complement deficiencies. So the complement, one very important thing what you need to write is hereditary angioneurotic edema. It is because of C1 esterase inhibitor. Second thing what you need to write, we already discussed this, C5 to C9 deficiency leads to, the membrane attack complex deficiency leads to recurrent infections with Neisseria. We already discussed while we are doing microbiology Neisseria topic. So this is one element, C5 to C9, recurrent infections with Neisseria species, and C1 esterase inhibitor is hereditary angioneurotic edema. Third important thing is factor H. Factor H deficiency leads to hemolytic uremia syndrome. Factor H deficiency leads to hemolytic uremia syndrome. And last but not the least, more important, the DAF and CD59 components of the complement, they lead to paroxysmal nocturnal hemoglobinuria. The PNH part is not important for your exam, but what happens in PNH is, as I already told you, the alternate pathway is there. Alternate pathway does not require any antibody formation. So it just requires a contact with an antigen, but it is under control, under check. In the body, every day, there is the alternative pathway going on, but it is under check and it is not killing our own body cells because it is controlled by these elements which are present on the cell surface, that is CD59 and DAF. If they are deficient, the active complement pathway, that is the alternate pathway, is always active in the body. It goes and it looks at the antigen. Sometimes it also looks at our own body cells. Our body cells needs a daily protection against this alternate pathway. How do they protect against themselves? By possessing these two cell surface proteins, that is DAF and CD59. So whenever the alternate pathway C3A comes here, and whenever it looks at the cell, if these two components are present, it leaves them or spares them. But what happens in patients with PNH, there is an absolute deficiency of DAF and CD59, and this starts attacking the cells. Because the RBC has like abundant amounts of these complement protecting factors, in patients with paroxysmal nocturnal hemoglobinuria, mainly the RBCs are affected. But all the cells will be affected, but RBCs are most severely affected. That's the reason patient will have anemia. So what are all the 4 things you're taking home? One is C1 esterase inhibitor, hereditary angioneuritic edema. Factor H is hemolytic uremic syndrome. Membrane attack complex is Neisseria gonorrhea, meningococcal infections. And DAF and CD59 is PNH. So what are the things that we are going to discuss in hypersensitivity reactions? First thing, I'll give you, as usual, the definition, okay, and also the causes, followed by the mechanism of each hypersensitivity, like how it develops, the pathogenesis part for type 1, type 2, type 3, and type 4. You need to remember the flow chart. That's more than enough for exam. If you want to draw a diagram, you have to draw a hand diagram, easy diagram for this. And I'll give you Gell and Coombs classification of hypersensitivity reactions. I'm trying to put all of the components together so that it is easy for you to revise at the end of the day. And I'll also give you the typical manifestations associated with hypersensitivity reactions. And I'll also add a note on type 5 and type 6 hypersensitivity reactions. So this is how this discussion of hypersensitivity goes. We're starting with the hypersensitivity reaction definition. Normally, the inflammatory reaction in the body, whenever there is an immune response, so there are 2 things that occur. The body will have an immune response, and immune response are of 2 types. One is humoral mediated and other is cell mediated immunity. Okay, body will produce these kind of responses. Whereas inflammation, we have an acute inflammation or a chronic inflammation. We have an acute or we have a chronic inflammation. These are 2 things that are happening. What do they do? They protect our body against any foreign invasion. But if there is loss of control, or if there is an uncontrolled response, and if there is an improper response by these to protective phenomenon, and this inflammation now becomes chronic, and there will be inflammatory disorders, chronic inflammatory disorders which are occurring in the body because the inflammation is not under check. Similarly, there will be some diseases in which there is an irregulated or unregulated immune response. So beyond the point, the response goes on and it damages the body. So these are the hypersensitivity reactions. So injurious immune reactions are hypersensitivity reactions, and the diseases which are resulting from injurious immune reactions are hypersensitivity diseases. Normal immune reactions are protective, but here we have injurious immune reactions. So that's the basic definition for hypersensitivity reactions, and they are classified into 4 types based on the immune mechanism. We'll be discussing the immune mechanism of each hypersensitivity. So the first 3 are the immune response which is dysregulated or which is causing injury. For the type 1 to 3 responses is humoral mediated, that is antibody mediated injury. Whereas in type 4, it is antibody independent, so it doesn't require antibody to produce injury. So this is cell mediated and it is basically of T cell. because antibodies are produced by B cell. These are all B cell dependent, whereas the cell mediated immunity is a T cell dependent immunity. So what are the causes for this hypersensitivity reaction? Sometimes the autoimmune reactions will trigger it. Sometimes any microbial antigen which is entering into the body triggers it, and also the environmental antigens. You take the example of any pollen grain which can cause atopy, and any drug which can cause anaphylactic kind of reaction. These are reaction against environmental antigens. Reaction against microbial antigens, you take hay fever, farmer's lung, which are type 4 hypersensitivity, tuberculosis, there will be some type 4 hypersensitivity component and cell mediated immunity component. All these are reactions against microbial antigens. So these are the causes in nutshell. They're not asking causes, but just for the sake of idea, I'm putting it there. So let's go to the next part, that is the type 1 hypersensitivity mechanism. What are the three things you take home? Type 1 hypersensitivity means there is production of IgE antibody. So while we're discussing immunoglobulin, I already told you IgE is a main component for type 1 hypersensitivity. And what does it do? It causes immediate release of mediators, the chemical mediators, mainly the histamine, histamine from the mast cells. So whenever histamine is released, it is a chemotactic agent and it's a vasodilatory agent. And what does it do? It produces all the symptoms by recruitment of the inflammatory cells. So these three things, if you remember, it's well and good. But if you want to draw an image and write a detailed expression, as I told you, the cause of hypersensitivity is exposure to the environmental antigens. And here there is an exposure to the allergen or a pollen grain on the mucosal lining. And whenever there is an exposure to the allergen, the B cells, the B cells, which are the B type lymphocytes, which are freely floating, what do? The allergen goes and binds to this B cell. The allergen goes, the antigen goes and binds to this B cell. The antigen antibody complex binds to the B cell, and B cell, there is an activation of the T helper 2 response. There are two kinds of response, T helper 1 response and T helper 2 response. We'll be discussing about the different types of helper responses. But what you need to remember at this point of time is, whenever there is attachment of an allergen to the B cell, there will be a T helper 2 response which is getting activated. And whenever there is an activation of the T helper 2 response, there is transfer of or transformation of the B cell into the antibody secreting plasma cell. So whenever there is a formation of plasma cell, there is a release of immunoglobulin E into the body. And this immunoglobulin, whenever it is produced, as I already told you, what does it do? It goes and it binds to the mast cells. Don't worry about where it binds and all, but IgE goes and binds to the mast cell. And whenever there is an exposure for the second time, mast cell is now sensitized. It is sensitization phase. So whenever it is sensitized, whenever there is a repeat exposure to the allergen, to the same allergen, there will be a massive release of mediators from this activated mast cell. And these mediators are vasoactive amines, lipid cytokines and all. What to do? They cause an immediate hypersensitivity response. And what are the two important kinds of hypersensitivity response? The milder one is allergy, what we see usually in clinical practice, but anaphylaxis is a life-threatening one. So these are the two types of hypersensitivity reactions. So what are we discussing here now? We are discussing the mechanism of type 1 hypersensitivity. What you need to remember is IgE antibody, mast cell, release of histamine, vasodilation, and also the activation of the complement system, cytokines, vasoactive amines and all. Not complement pathway. It is more or less the release of histamine which causes all the symptoms. Next is type 2. And what to have in type 2, the mechanism. Type 2 has like almost three kinds of things. One thing, whenever an antigen enters into the body, whenever there's an antigen entry into the body, the antibody binds to it, and there will be the phagocytosis, or there will be the lysis of the target cell, So these are all there will be antibody mediated cytotoxicity. or cellular dysfunction. Let's see these three in detail. But before going into the details of the picture, for you, if you want to remember type 2 hypersensitivity, just remember this flowchart. There will be production of antibodies IgG and IgM, and they bind to the antigen on target cells or the tissue. So it's a fixed antigen here. Okay, the antibodies which are produced in the body, they are binding to a fixed antigen. So what are they doing? They're binding to the fixed antigen which is present on the cell. So why I'm stressing on this fixed antigen? When I tell you the examples for this type 2 hypersensitivity in the next part of the discussion, the basic difference between type 2 and type 3 is based on the mobility or fixity of the antigen part. In type 2, the antigen is fixed. In type 3, the antigen is mobile. In type 2, on the cell, there will be antigen. Antibody comes from the circulation, it binds to the cell and it causes all the reactions. But in type 3, what happens? Antigen is in the humoral cavity, antibody is also there free floating, they bind together, they form an antigen antibody complex, they migrate to a tissue, they'll get deposited there. Later on, they'll cause all the symptoms of type 3 hypersensitivity. So when I'm discussing hypersensitivity, it is not protective, it is injurious. Whatever happens with the immune system to protect the body, all those steps will be happening, but they are happening in an exaggerated manner wherein they cause injury to the body. So with this basic point in mind, let me tell you the type 2 hypersensitivity. So what is happening here? See, this is a phagocyte, and the phagocyte has both the FC receptor and also the C3b complement receptor. Okay, and this is an opsonized cell. Okay, this is the cell onto which you will see the antigens. So the cell has antigens bound to it. Now the antibodies are binding to it and they're binding to the phagocyte, and slowly the phagocyte is taking in the antigen. So this is how phagocytosis occurs. Okay, so this is one step. Next is, you just try to observe here. This is again the FC receptors, okay, which are present on the cell, which will identify the FC component of the antibody. And this is an antigen, which is a free floating antigen, which is bound to the surface. Okay, and what they're doing, there will be a complement pathway which is getting activated here. Okay, and the complement byproducts are C5a, C3a. What are these? These are all anaphylatoxins and chemotactic agents. And there will be attraction of the neutrophils, enzymes, reactive agents, intermediates, everything coming at a place to cause tissue injury. Okay, this is one part. Third part, how this antigen antibody reactions occur. See, antibody mediated cellular dysfunction. What is this? Antibody mediated cellular dysfunction. The antibody, what kind of type 2 hypersensitivity I am discussing here is two things. One is Graves' disease, and this is myasthenia gravis, the classic examples for type 2 hypersensitivity. So after this discussion, you will understand all three components in detail. But what I'm telling you is, in type 2 hypersensitivity, there are three mechanisms. One is activation of the phagocytosis, and this phagocytosis goes on, and if this reaction occurs, there will be some injury to the body. Similarly, there will be lysis of the target cells by activation of the complement pathway. And third important thing is antibody mediated cellular dysfunction. You know what happens in myasthenia gravis? Myasthenia gravis has production of acetylcholine receptor autoantibodies in the body. And what do? They go and bind to the neuronal plate and they'll destroy all these acetylcholine receptors. Okay, they bind to it and they'll not allow the released acetylcholine from the presynaptic vesicle to bind and produce a muscle contraction. So that's the reason the patient with myasthenia gravis will have a muscle paralysis or muscle weakness. Okay, the patient will have morely muscle weakness, which progresses from morning to evening, which becomes severe as the day progresses, right? So this is what is happening here. In these diseases, the antigen antibody is not damaging the tissue, or there is no phagocytosis. What they're actually doing is they are causing dysfunction of the cellular mechanism. What are they doing? They're causing dysfunction of cellular mechanism. So this is one type wherein here the antigen is fixed. That is the reason we are putting this in type 2 hypersensitivity category. Now the antibody is coming and binding to the surface, and it is not allowing the muscle to contract. What happens in Graves' disease? In Graves' disease, there is production of autoantibodies against the TSF receptor. And what do? When they bind, they'll not damage the cell. Rather, they stimulate this receptor. Usually, whenever TSH comes, the TSH are thyroid stimulating hormone. Whenever it comes and binds to the TSH receptor of thyroid cell, it carries a message. And in conditions wherein there is hypothyroidism, the TSH levels in the body, they'll increase. Why do they increase? So that the message goes to the thyroid cells and there will be release of thyroid hormones. But what is happening in the Graves' disease? Though the thyroid levels in the body are normal, still because of the production of these autoantibodies against TSH receptor, they are binding and they're giving a false signal of deficiency, and actually the antibody is stimulating the receptor without the hormone. Okay, so I'm saying dysfunction, I'm not saying malfunction or decreased function. There will be some stimulation or there will be some decrease in function. So these two are antibody mediated cellular dysfunction, classic examples, which is a part of type 2 hypersensitivity. So we're done with the type 1 IgE mediated. Type 2 is either IgG or IgM, and we'll be having phagocytosis, complement lysis, or the antibody mediated cellular dysfunction. Mention this in the exam. So hope after this session you have a clarity about what is antibody mediated cellular dysfunction and what kind of hypersensitivity it is. So let us quickly move to the type 3 hypersensitivity. So the same thing, remembering this much is more than enough. Anyway, you have an image, but here the difference is it is a mobile antigen. It's not a fixed antigen. So there will be antigen antibody complex formation, and there will be, they go and they deposit in the tissue. This leads to complement activation in the tissue. Whenever complement gets activated, there will be recruitment of leukocytes. Everything, as we already discussed, associated with complement. There will be release of enzymes and toxic molecules and local damage. And this type 3 hypersensitivity is seen in conditions like serum sickness and also most of the autoimmune vasculitic disorders. Okay, mainly the Childs-Cross syndrome. There are so many autoimmune vasculitic syndromes like even the same concept applies to Sjogren's syndrome, same complex applies to SLE, more important. These are all type 3. And what happens in these, there will be an autoantibody produced in the body against an antigen, and these antigen antibody, they go and they land in the vessel, blood vessel. So there will be activation of the complement. So there will be damage of the vessel, and blood vessel inflammation is known as vasculitis. That is the reason in most of these diseases, systemic vasculitis is seen. So that is what is depicted in the picture here. There is an antigen in the circulation, and there is an antigen antibody complex formation. And later what is happening? They are getting deposited in the vessel wall. Okay, and complement is coming and it is bringing in neutrophils, and there is damage which is starting here because of the neutrophil. And to correct that, the platelets are aggregating there, they are forming thrombus, and the thrombosis will produce more and more tissue complications. So this is the other name for type 3 hypersensitivity is immune complex mediated hypersensitivity. So it is immune complex mediated hypersensitivity. Next is type 4. Type 4, all the type 1, 2, 3 are there dependent on antibodies. This is antibody independent or weakly dependent. Let's not confuse ourselves. Let's put it as antibody independent reaction, or it's a cell mediated type hypersensitivity. Here there is no antigen antibody complex. Here the major attack is by the activated T lymphocytes. Somehow, because of an inflicting agent or an inciting agent, there is activation of the T lymphocytes in the body, And these activated T lymphocytes, they're causing release of the cytokines in the body. Okay, this is not complement pathway. In type 2 and type 3, you know, the complement pathway is playing a major role. But here, there is release of cytokines, the chemical mediators, and they cause the inflammation and also the activation of the macrophages. What do they all do? They produce a T cell mediated cytotoxicity leading to the death or injury to the cell. Okay, so see here, this is an antigen presenting cell carrying an antigen with the help of MHC complex to the CD4 T cell. Okay, and whenever the CD4 T cell has a T cell receptor and T cell receptor is binding to MHC complex, okay, and whenever they both bind, they lead to reactions, a cascade of reactions wherein there is release of cytokines. Okay, it's all cell mediated immunity, and these cytokines has different functions. They cause vasodilation, some cause vasoconstriction, they cause fever, and they also are some are pro-inflammatory, some are anti-inflammatory. But here, more or less pro-inflammatory compounds are released, and there is an inflammation which leads to tissue injury. There is an inflammation which leads to tissue injury. Sometimes what happens, see, there is a cytokine mediated is one. In some cases, the soluble mediators will not come into the picture. Directly, the T cell, the cytotoxic T cells, that is CD8 cells, CD4 cells, they take help. Okay, they require help of cytokines to produce all the tissue injury component. But CD8 cells, see, this is a normal cell, normal tissue on which there is an attack. Okay, there is an attack. So whenever there is an attack, the antigen presenting cell takes this whatever antigen is there and it is presenting to CD4 cell. But this cell itself will have MHC complex, MHC1. Okay, and this complex is MHC2. The cell itself will have MHC1, whereas antigen presenting cells will have MHC2. Now, CD4 cell only binds to MHC2. That is the reason it is going to antigen presenting cell. But CD8 are cytotoxic lymphocytes. They directly go to the site of war. This is the viral infection, and this is the MHC2. They directly check the antigen from the cell itself, and they directly cause cell injury. So this is the T cell mediated cytolysis or cytotoxicity. Both the things occur, one with CD4 response and other with CD8 response, and these two are the cell mediated response. If they are aggravated, if they are severe, they'll produce type 4 hypersensitivity. So we're done with the mechanism. Type 1 IgE, type 2 IgG IgM, and type 3 is antigen antibody complex mediated, type 4 is cell mediated hypersensitivity. So this classification will summarize our discussion. There are two, Gell and Coombs classification of hypersensitivity reaction. We have type 1, 3, 4. This thing done, humoral, this is cell mediated done, and the response in this is immediate because there is a humoral factor, because plasma cells, they are ready to deliver antibodies whenever needed. So that's the thing, reaction is immediate. But type 4, the reaction is delayed. So the timing and all not important, but more important thing is the antigen is soluble here. Here it is soluble. Sometimes CD4 mediated, it should be soluble, and CD8 it is bound. That's the reason it's directly coming there. But cell surface bound is type 2, as we already discussed. And the mediator which is involved is IgE here, IgG here, antigen antibody complex, and it is T helper cells. Next, what is the effective mechanism? This is mast cell degranulation, and this is antigen dependent dysfunction or cell mediated cytotoxicity, and also complement mediated. There will be complement activation and inflammatory response. You are aware of all these things because we discussed in detail, and that's the reason I'm going fast. And this is macrophage activation and cell mediated cytotoxicity. Rest of all, this desensitization is not important for exam, just leave it. What you need to remember is like the examples they're asking, what they're doing, define type 1 hypersensitivity and explain the mechanism of anaphylaxis, or explain the mechanism of asthma, or explain the mechanism of allergy, or explain the mechanism of atopic dermatitis. Whatever they're asked, you know the mechanism, right? And next is type 2. If they ask RH incompatibility, ABO incompatibility, transfusion reactions, Graves disease, hemolytic anemia, autoimmune hemolytic anemia, all these are like the fixed antigen is getting reacted with a soluble antibody. So you can write the mechanism there. Type 3 is Arthus reaction, serum sick reaction, some glomerulonephritis, rheumatoid arthritis. All these are type 3 examples. Type 4 examples are tuberculin test, granuloma formation in TB, contact dermatitis is one, because see, this is very important, because in dermatology, dermatitis is an important topic. Okay, the atopic part of dermatitis is humoral mediated, but contact dermatitis, if they ask question, when I'm correcting papers, most of them are writing that it's a type 1 hypersensitivity. No, it's a type 4, or contact dermatitis is type 4 hypersensitivity reaction. So this will conclude our discussion on hypersensitivity reactions, but it may not be important for your exam, but just for the sake of completion, there are two more types of hypersensitivity, but now they removed it and they included with the old hypersensitivity. One is the stimulatory hypersensitivity. Graves disease, as I already told you, right? In most of the hypersensitivity, there is damage to the tissue, or there is autobody mediated cellular dysfunction. But what is happening with Graves disease? There is actually a stimulation. The receptor antibodies which are produced, they are stimulating thyroid cell to release the thyroid hormone. So this kind of a stimulatory hypersensitivity is rare to see, and it is seen in Graves disease, and that's the reason you can call it as type 5 hypersensitivity. But nowadays the literature has removed this. They have included with type 2. Next is type 6. It is again included with type 2. What happens here is it's an antibody dependent cell mediated cytotoxicity. The same thing as I already told you, it's a immune defense mechanism, but this is mediated by, more important thing, natural killer cells. So when I discuss natural killer cells in the next session, you will understand it in a better way. But for you now, the take home is 5 is Graves disease, 6 is ADCC. Just remember this, because the ADCC question was asked once in the mains, okay, in medical science optional exam. They asked define antigen dependent cell mediated cytotoxicity and add a note on natural killer cells. So we'll be discussing that in detail. Now let's go to next topic called immunological tolerance and mechanisms of autoimmunity. So the next part of the class, the main concept here is immunological tolerance. And if you understand how the body is able to detect our own cells as their own signature, and how about our body not producing any attack against the self antigens. So what are all the protective phenomena which are present in our own body? If you know them, if there is any disturbance to this natural or our own tolerance, then there will be production of autoimmunity. So understanding immunological tolerance and understanding mechanism of autoimmunity, they both go hand in hand, because if you know one concept, you can apply the same to the other concept. So in the first part of the discussion, try to focus here. What is immunological tolerance? It is a state in which an individual is incapable of developing an immune response against his own tissue antigen. Simple, the definition. What is immunological tolerance? But what are the mechanisms of immunological tolerance in the body? One is central and other is peripheral. We all know we have central lymphoid organs and the peripheral organs. Central lymphoid organs are mainly the thymus and bone marrow. The T cells, they mature inside the thymus. That is the reason they are called as T cells. Whereas the B lymphocyte, they mature inside the bone marrow. That is the reason they are called as B cells. So with this basic knowledge, let me tell you what happens with central tolerance. Whenever within the thymus or the bone marrow, if there are any self-reactive T cells or self-reactive B cells which are produced anonymously, if they are present, the thymus and bone marrow, they act and they kill them. They remove these self-reacting cells either by negative selection, that means they only select the non-self reactive ones, and also sometimes they do some receptor editing so that all this self reactive are again made non-self reactive. So these are the two mechanisms by which they act. One is negative selection. Let's not go into the details how this happens and all. It's not important. But for exam point of view, right, central tolerance, where it occurs, and what are the mechanisms by which occur. One is negative selection and other is receptor editing. So this will conclude the central tolerance part. The peripheral tolerance is more important because most of the times it is the disturbance in this peripheral immunological tolerance that will lead to the autoimmunity. There are several backup mechanisms which are occurring at the tissue level so that they can counteract these self-reactive T cells and escape the central tolerance. So what are these? 1, 2, 3, 4, 5, 6, 7. There are 7 techniques or 7 mechanisms which you need to remember. First one is just ignore, because it's an ignorance. The self-reactive T cells, they do not either encounter the self-antigen or they ignore it. Two cells. See, if there is a self-reactive T cell, it may not see a self-antigen. Even if it sees the self-antigen, it will ignore it. So both are same. So ignoring the self-antigen even after recognizing it is a state of ignorance, and most of the self-reactive T cells in the body, they are in a state of ignorance. These points I am repeating them again. Yeah. So we're starting with peripheral tolerance. The self-reactive T cells might never encounter the self-antigen which they recognize, and therefore they remain in a state of ignorance. This is one thing. Next is energy. Okay, energy is no response. It's similar to ignorance. Ignorance is they're not experiencing or encountering the self-antigen again in their life. But energy is even after by looking at the self-reactive antigen. So this is self-reactive T cell and this is self-reactive antigen. Even after getting in contact, the response is not happening because the co-stimulatory signals which actually help in activation of this self-reactive T cell, they're getting blocked. So what are the co-stimulator signals? The binding of this is if this is antigen presenting cell, okay, it has MHC molecule 2 in it, and if this is the antigen and here is a T cell receptor and this is a self-reactive T cell, the binding of these three is not enough. There are some co-stimulatory molecules, that is, one is very important is B7 molecule which is present on APC. Another co-stimulatory molecule is CTLA4 molecule which is present on the T lymphocyte. So whenever they interact, they produce some co-stimulatory molecules so that this reaction can happen. So now the self-reactive and T cell and APC, they are in contact, but the co-stimulation is not happening between the B7 of APC and CTLA4 of self-reactive T cell. So this is what is energy, blocking of the co-stimulatory signals. The third mechanism by which there is peripheral tolerance is phenotypic skewing. Okay, so most of the times, as we discussed, the cell-mediated immunity, which is either because of the CD4 or because of the CD8. In CD8, it is directly causing tissue toxicity by perforin release. But in CD4, what is happening? CD4, there is a mediation of cytokines, and these cytokines are the major cause of tissue damage. Okay, there is a self-reactive T cell, which is a CD4, and there is an APC, and when they bind together, there should be release of cytokines which can damage. But here, the self-reactive T cells, they are secreting non-pathogenic cytokines. So whatever cytokines we have released, they are not either vasodilatory, vasoconstrictory, or they are not eliciting any inflammation or any dangerous inflammation. So this is phenotypic skewing. There is a switch in the skewing of the phenotype of the cytokines which are produced. Okay, so try to understand these in detail, because at the end of the day, when you want to mug them up, you can't mug up because these are all tough words. If you understand, at least you can write. You can write what is ignorance, Non-encountering self-antigen, you can write energy, blocking of the co-stimulator signal. Now you can write phenotypic skewing because the cytokines which are produced are now non-pathogenic. Next is apoptosis. So what happens in apoptosis? Uh, the self-reactive T cells, okay, they undergo death. Okay, so if this is a self-reactive T cell and if this is an APC, if there is an interaction between these two, there is a apoptosis signal released with this interaction, so that the self-reactive tissue is gone. Fifth very important thing is release of regulatory T cells. In our body, there are some regulatory T cells, and what is their function? Whenever there are self-reactive T cells in the body, the regulatory T cells will take over. They'll not allow the autoimmunity to develop. So this is one important thing. Last two, let's just focus on them. Dendritic cells, which are present in the body, some are immature forms and some forms are tolerogenic forms. Okay, dendritic cells are, some are immature and some are tolerogenic. Their function is to capture the self antigens, because dendritic cells are a variety of antigen presenting cells. What do? They capture self antigen, and after capturing, they process it. Okay, after capturing, they process it and they down regulate the expression of the co-stimulatory ligands. We already discussed co-stimulatory ligands. One is B7 and also other one is CD40. Okay, so this is one thing. Or they also help in production of the regulatory cells. Let me go back to again. What are dendritic cells? Dendritic cells are antigen presenting cells, and there are two varieties. One is immature D cells and other is tolerogenic D cells. What do they do? This category of dendritic cells, whenever they act more on self antigen. So this is the self antigen. They detect the self antigen, they take this self antigen inside. Now what they do? They do two functions. One is production of regulatory T cells. Regulatory T cells, what do? They down regulate self reactive T cells. The other function is down regulation of expression of the co-stimulatory ligands. So CD40 and B7 molecules are down regulated. So they're doing two functions. One, they are producing energy. Other, they are producing more regulatory T cells. So this is the function of dendritic DCs which are present in the body. Last, very important, some of the self antigens during our embryological time or embryological development, like testicular antigens and also lenticular or lens antigens, they are tightly packed, okay, and they're securely packed so that they're never exposed to our tolerance system. So we are actually sequestering these self antigens, and the areas where we are sequestering these, we call them as immunologically privileged sites. Okay, these immunologically privileged sites, they protect against autoimmunity. Okay, and what happens, like you take corneal antigens or lenticular antigens and testicular antigens, if there is breach in these immunologically privileged sites, let's say there is damage to one eye, there is release of lenticular protein, what happens? The other side will also get affected after few days, because there is a loss of this peripheral tolerance, that is sequestered antigens are now released, the body will mount an autoimmune attack. So that condition wherein there is an eye damage to one eye which is affecting the other eye, this condition is known as sympathetic ophthalmitis. Okay, this is because of the loss of this immunologically privileged sites, there is damage. Similarly, some spontaneous infertility may be because of the damage to the testicle and the release of these self antigens, so that there will be production of autoantibodies against the sperm cells, leading to infertility. So all these are the concepts here. So now you have a clear idea what are all the backup mechanisms that prevent peripheral, that cause peripheral tolerance: ignorance, energy, phenotypic skewing, apoptosis, regulatory T cell production, dendritic cell production, and more important, last is the sequestration of self antigens. So hope you have a clear idea of how we are protected against autoimmunity. So the same thing is depicted here in the in the image. This is the peripheral tolerance, and this is the central tolerance, which is occurring in the thymus and the bone marrow. Okay, so this is occurring because of the deletion or negative selection or receptor editing. But here only the deletion part which we discussed, and these are the energy part which we already discussed. There are few more things. There is release of regulatory T cells and all. Just go through this in the notes. Focus on this. Now the most important part. What are the mechanisms of autoimmunity? If I read them now, just you will understand. Do you remember the apoptosis induced cell death? Whenever there is activation, there is induced cell death. Apoptosis part. If the apoptosis part is failed, this is predominantly seen in SLE. What I am telling you now is, what is that failure thing which is causing autoimmunity? That is what is the mechanism of autoimmunity and some of the examples for it. First thing is failure of apoptosis. Second, loss of T regulatory cells. It occurs in most of the autoimmune diseases. That's why the reason I hadn't mentioned any examples. There will be some loss of T regulatory cells. Breakdown of CTLA4 mediated T cell energy. Do you remember the T cell energy? We got two things: B7, which is present on APC, CTLA4 is present on the T cell. So this CTLA component, the co-stimulatory, if it is not able to down-regulate, in that cases you will see multiple sclerosis, rheumatoid arthritis, or psoriasis. So the breakdown of CTLA4 mediated T cell energy, which is a co-stimulatory molecule present on the T cell. We're done with three things. You know the mechanism. Fourth, simple, very easy. Release of sequestered antigens. I already told you the spermatozoa and ocular antigens. Due to the injury to these organs, we'll see the sympathetic ophthalmitis and spontaneous infertility. Very easy. Next, molecular mimicry. Do you remember why there is thrombocytopenia in dengue? The NS1 antigen component of the dengue virus has a structural similarity to the antigen which is present on the platelets. So that's the reason the body antibodies, they're killing dengue, but they're also killing the platelets, leading to thrombocytopenia. So this is molecular mimicry. We also see the same in acute rheumatic fever. This is bread and butter for us, right? While we read the rheumatic fever, it is because of the molecular mimicry. And also the post-streptococcal glomerulonephritis. These are the two important conditions wherein there is the molecular mimicry leading to autoimmunity, leading to the damage either to the heart or to the kidney. And third example, now you can write the dengue. Dengue-induced thrombocytopenia is because of the molecular mimicry. So fine, five important mechanisms. If you write in the exam, you'll get full marks. But let us also go into a bit details, okay? Because those are all easy. The next four are also important. Sometimes the autoimmunity may be mediated by the super antigens in the body. Do you know TSS, toxic shock syndromes? Usage of vaginal tampons leads to the development of the toxic shock syndrome. And we don't know why a staphylococcal infection is actually causing the toxic shock syndrome. The reason is the antigen is so powerful. Okay, the antigen is so powerful. It is activating so many T cells, leading to a massive cytokine release and damage to the body. Okay, so this kind of polyclonal lymphocyte activation can also be an autoimmune condition wherein there is a production of toxic shock. Okay, and one more mechanism of autoimmunity is exposure of cryptic self-epitopes. On the antigen presenting cell, there are some epitopes. Okay, and these are called dominant episodes. Okay, and these dominant episodes, what do they do? APC, the dominant epitopes, they prevent autoimmunity. Their main function is prevention of autoimmunity. But when their count decreases, there will be the minor variant called the cryptic self-epitope, which function is to promote the growth of self-reactive T cells. These are cryptic self-epitopes. This concept is not important for you, forget that. But why I'm telling you is, to remember this also, you need to know what is the cryptic self-epitopes. Cryptic self-epitopes means normal self-epitopes on the APC will prevent the development of autoimmunity. If the normal or dominant variety of epitopes are coming down, then the cryptic, the hidden epitopes will dominate. That will lead to promotion of the autoimmunity. So that is multiple sclerosis. Similarly, epitope spreading. The normal epitopes which are actually preventing, now there is a development of immune response to the same epitopes which are actually protective. Okay, as there is a release of self antigens in the body. So this is epitope spreading. So you need to remember the exposure of cryptic self epitopes, epitope spreading, and also bystander activation. Activation of T cells specific for an antigen X during an immune response against Y. Actually, the T cells are produced to fight this Y antigen, but we don't know why. But they're going and acting against X antigen. So this is known as bystander activation. Though the T cell is not for this X antigen, it is acting against this X antigen. So this bystander activation is a principle seen in viral infections. So these are all the mechanisms of immunity. Let me revise them for you again. Breakdown of energy, failure of activation induced cell death, loss of T regulatory cells, release of sequestered antigens, molecular mimicry, polyclonal lymphocytic activation, exposure of cryptic self epitopes, epitope spreading, and bystander activation. Try to remember the example because they'll give you more weightage. So what is autoimmunity? You have a clarity, but to write it in words, autoimmunity is a condition in which the body's own immunologically competent T cells or antibodies, they act against its self antigens, resulting in both structural or functional damage. So this is autoimmunity. These are all the immune function and what is happening. How to assess all these immune functions? Like by doing either history and examination, because they may ask five mark question. They're giving immunity in the syllabus, and somehow the 10% of the questions they ask like a bouncer kind of things or new kind of things. So in that they may ask, how do you assess immune status of a body? What are all the techniques available to assess the immune status of a body? Very important thing is history and examination. Okay, by asking history about any occurrence of the recurrent infections in the body, okay, or recurrent skin lesions, okay, everything comes to the history part. Examination, you have to look at the lymphadenopathy and examination of the lympho-reticular system. That's fine. But investigation part, we look at CBP, complete blood picture, wherein you will see DLC and you look at the count of the lymphocytic count. Okay, you look at the lymphocytic count in CBP, and also the evaluation of immunoglobulins, because in immunodeficiency diseases, you will see low levels of selective or either all immunoglobulins based on various genetic conditions. And you can also assess the B cell function. B cells, they help in production of immunoglobulins. So if you look at the naturally occurring antibodies against rubella, influenza, tetanus, diphtheria, you measure them. If there is a natural antibody protection, so the B cell function is normal. Okay, and you can also assess B cell function by assessing the response to immunization. We are giving polio, HBV, typhoid, and all those things. We are giving an inactivated or a killed antigen so that the body will produce antibodies. So antibody production is a function of B cell. So this is how, either by vaccination or either by natural infection, you can assess the B cell function. So how do you assess the T cell function? The T cell function is cell mediated one. So we have tests, right? The Montauk's test, the Lepromant test. Okay, all these skin related tests wherein we inject a protein purified derivative. So we assess T cell function in that way. Okay, and the T cell function can also be assessed by looking at the thymic shadow by doing a chest X-ray in children. Okay, so because in some patients there is a Dijos syndrome, you will see thymic aplasia. Okay, thymic aplasia. So in those cases, you have to do chest X-ray to look at the lack of thymic shadow in a child. And complement assess, yes, there are some conditions in which you will see complement deficiencies. I already explained you all the complement deficiencies. So if sometimes now these complement stuff and all is tough to do, it requires expertise and it's a sensitive lab and it's a costly procedure. That's the reason instead of doing a individual complement assay, most of the times we do total hemolytic complement CH50 test. Somehow in the exam, if they say CH50 assay has showed low scores and all, don't worry, what is this CH50? It's a complement hemolytic component. Okay, so we do this test for complement assay. Next is, if you want to look at the phagocytic function, what is the test which is available? NBT test. And NBT test is used to assess phagocytic function, that too mainly for chronic granulomatous disease, which is because of a deficiency of NADPH oxidase. Okay, so this NBT test will produce a tetrazolium, which is the tetrazolium will produce a blue colored compound. When we do it, the blue colored compound is produced only when there is an active NADPH oxidase, because the conversion from normal yellow color of NBT to blue color occurs only presence of NADPH oxidase. What happens in this CGD, that is chronic granulomatous disease, this NADPH oxidase, the respiratory burst mechanism, the radical producing mechanism, which helps in damaging the infections, so this is deficient. So this leads to non-production of blue color. Okay, so if there is a blue color, NBT is positive, that means it's a normal individual. If it remains yellow only, so there will be a deficiency of NADPH oxidase. So NBT is negative in case of chronic granulomatous disease. So this is one part of assessment of immune function. If they ask and if you write this, it will impress the examiner. And next, you can also do lymphocyte cell cultures, okay, and lymphokine assays. Just remember those names. Or also you can do, see, this is nowadays, this is very important, because we are in an era of transplantation medicine. Okay, transplantations are now a day in day out procedures. Okay, so many transplantations, kidney, liver and all, and hematopoietic stem cell transplantations. The surgery rate is increasing. Okay, so when we do a transplantation, the major issue is the graft rejection. So that's the reason we match both donor and recipient. How do we match both donor and recipient? With the help of mixed lymphocyte cultures. So when you are assessing immune function, the mixed lymphocyte culture is done. It is used for the compatibility testing in transplant work. It is used for compatibility testing in transplant work. What do we do in one set? There are two sets, set 1, set 2. Okay, this is donor and recipient. So set 1 is donor, set 2 is recipient. And when we mix them, if there is a blastogenic response, that means if there is a response or transformation is happening, the transformation is because of the antigenic discrepancy between the two cell sets. Okay, so this may later lead to the graft rejection or graft versus host disease, and we'll discard that donor. So this is how the mixed lymphocyte cultures are done. So your take home is assessment of immune function. You have to remember these 8 important tests wherein we assess immune function. Last year, they asked this important concept called monoclonal antibody. And when it comes to pharmacology or microbiology, the monoclonal antibody questions are a trend now, even in postgraduate entrance examinations. And for medical sciences is optional, not completely, but there is a slight shift in trend from asking traditional questions. They ask the questions from the syllabus itself, but they go to an extent wherein you won't be touching those concepts, because last time monoclonal antibody is an important question. So for the last 10 years, they're asking classify immunoglobulins. Suddenly what they did, they shifted to, from the same immunoglobulin, they have shifted to the monoclonal antibody, but it's also an antibody. It's also an immunoglobulin, but it's not polyclonal, because polyclonal are what naturally produced in the body, but monoclonal is an experimental. It's an iatrogenically produced antibodies. They help in research, they help in production of the compounds. They have a vital role nowadays. They help in treatment of the malignancies, the monoclonal antibodies. They're blossoming like anything, because yesterday, like we were discussing about the prophylaxis of the migraine, and there is CGRP peptide which is causing migraine, and there are monoclonal antibodies launched in the market to fight against these CGRP antibodies, and you can use this monoclonal antibody for prophylaxis of the migraine. See where the science is going. So because of the rapid development in the department of monoclonal antibody, nowadays they're asking questions from this. It's fine, monoclonal antibody, like whatever I have collected the details, you will know the definition, you will know the difference between monoclonal antibody and polyclonal antibody. You'll know what are all the latest monoclonal drugs and their indications. But more important thing is how they are produced, because the tools in biochemistry, the biotechniques, bioengineering techniques are also a topic of interest for UPSC examiner. So I'll focus more now on the hybridoma technology, which helps in production of monoclonal antibodies. And I had seen some questions in previous years. They, instead of asking about radio immunoassay, they'll give the names of the scientists and they'll ask, like, write about the procedure invented or why they got Nobel Prize, what is the specialty and all. So in that perspective, okay, it may look odd, but try to remember this Kohler and Milston are the ones who have developed this ingenious method for large-scale production of monoclonal antibodies, and they are awarded a Nobel Prize for Medicine in 1984. So what are these monoclonal antibodies? The antibodies which are produced by infection in the body, they have multiple epitopes or antigenic determinants, okay? And they go and stimulate separate clones of lymphocytes based on the epitopic predictability. And when it happens, the anti-sera from the body has immunoglobulins of different classes which act against different epitopes of the antigen. It's all a polyclonal response. So much is happening without any specificity. But if you have an antibody which is produced by a single clone and it has a single antigenic determinant, it goes and it only acts on that part and it only downregulates that part and it only helps in stopping or acting against that disease. This is known as monoclonal antibodies. So they are highly helpful in both diagnostic and research techniques and also the therapeutic. We're missing important point here. They are used in therapeutics nowadays, the monoclonal antibodies. So the diagram here on the right side is the hybridoma technology, which is used in production of these compounds. How we do it, let me tell you. This is the mouse, and we challenge it with an antigen for which you need antibodies. So you take that antigen for which you need an antibody so that you will use this antibody against this antigen at a later for treatment part. So you infuse this antigen into this mouth and you collect the spleen cells, because the spleen cells, now the lymphocytes part, spleen cells will have the lymphocytes, right? You collect them, okay, B lymphocytes. These are the splenic B lymphocytes and their function is production of antibodies. And now they are primed. Primed means they are sensitized to produce antibody which is specific to the antigen which I ingested. Now, similarly, you take myeloma cells. Myeloma means not multiple myeloma. Multiple myeloma is a disease wherein you will see monoclonal antibody production. That too, the light chains. Okay. This is specific. The multiple myeloma is a specific disease wherein the whole of the body, whenever there is an infection or cancer, the whole of the body will react in a polyclonal manner. But multiple myeloma is a monoclonal response. So that's the reason if I have myeloma cells means cancer cells. They produce only one type of the antibody. That's the reason they are called monoclonal cells. So why we are collecting myeloma cells? For two things: their mortality, because they are cancer cells, they won't die, they multiply. Similarly, one more thing what we are doing here is, for myeloma cells, one more property which is more important for is monoclonal cell production. But while you take this myeloma cells, the prerequisite condition is they should be HGPRT gene deficient. What is this HGPRT gene? It's a alternative pathway of nucleic acid production. We have a salvage pathway in nucleic acid metabolism. In our body, DNA and RNA are produced in a normal pathway, but when normal pathway fails, we have a salvage pathway wherein we produce nucleic acid like DNA and RNA. But here, you are taking myeloma cells which are deficient and which cannot multiply if there is a HGPRT in the medium. You cannot multiply if there is any HGPRT in the medium. You're taking this. Why we are taking this, I'll tell you. But we have started, we want to produce antibodies against this antigen named X. We have ingested X into mouse, and the mouse spleen cells will now contain B lymphocytes which can produce antibody against this X. Now you're fusing this with a myeloma cell which has an immortal property which can only help in growth of single type of monoclonal cells and which is HGPRT deficient. You don't know why this, I'll tell you that, but you know the first step of basics and all these things, now you know. Let's go to the next thing. So for this fusion, you can use electrofusion or polyethylene glycol, which is a chemical fusion. So you're mixing B cells, the splenic B lymphocytes, with the myeloma cells with the help of electrofusion. Now whatever cell which is formed is hybridoma. So hybridoma is a mix of B cell from the mouse plus the myeloma cell which we have taken. When they fuse together, they have a property like this. They are motile, they produce monoclonal antibodies, they have salvage pathway intact. Why salvage pathway is intact? Because the B cell has HGPRT gene. Salvage pathway intact. Now I'm collecting these hybridoma cells and I'm culturing them in HAT medium. After 10 to 14 days, I am able to get monoclonal antibodies. Basic procedure is fine, but let's focus on why this salvage pathway is important in the next part of the diagram. See, this HAT medium has aminopterin in it. HAT means hypoxanthine aminopterin medium. What is this aminopterin? It is a nucleotide synthesis pathway blocker. So it won't allow nucleoside synthesis to occur inside the medium. When you have unfused myeloma cells, they die here because, why do they die here? Because they don't have HGPRT, and in turn, you're putting a blocker. And unfused B cells also die in the medium because they have a short half-life. But fused B cell, because of the immortal property given by the myeloma cell, it will replicate. So the hybridomas will replicate. And because of the presence of HGPRT, though there is an aminopterin blocker in the medium, the body itself will produce a salvage stuff. Okay, salvage pathway gets activated, normal pathway is disturbed, and there is the only clonal proliferation of hybridoma cells. Because of the protection we have taken, unfused B cells, they die. Myeloma cells which are not fused, they will also die. Only you will get hybridoma cells, and these hybridoma cells are specific with desired antibody production. So this is hybridoma technology or monoclonal antibody production by these hybridomas. Hi doctor, this video contains information about what are the different monoclonal antibodies, what is the difference between a monoclonal antibody and a polyclonal antibody. Let us begin now. Monoclonal antibodies, we short form them by the symbol given here, that is MAB or MOAB, are identical immunoglobulins generated from a single B cell clone. Whenever there is an antigenic exposure, let us say we are vaccinating a mouse, okay, and we are injecting an antigen. Whenever there is an antigen exposure, the lymphoreticular system, it releases antibodies, right? Let's take an example of spleen. So it releases antibodies. These antibodies, they are not identical because they react with the same antigen but not on the same epitope. Epitope is an antigenic determinant, that is a signature on which antibody goes and acts. So these antibodies can bind to any epitope which is given here. So this kind of response is known as polyclonal response because it is binding to different epitopes of the same antigen. But if you take special precautions, if you perform a special techniques called hybridoma techniques, wherein you take those antibodies which are specifically binding to a single epitope, that is a particular epitope. So this response is monoclonal response, that is you are collecting identical immunoglobulins generated from a single B cell clone. So these are monoclonal antibodies. So that is what I am trying to explain. These antibodies, they recognize unique epitopes or binding sites on a single antigen. We do have an image I'll show you so that you will have a better understanding of this. And the next point is they are derived from a single B cell clone. Okay. And they target only single epitope. That is the feature which differentiates monoclonal from polyclonal as we discussed. And this is how we synthesize it. Like if you immunize an animal with an antigen, the B cell response is polyclonal. Then you select one particular B cell clone of your choice and expand them to make a stock of monoclonal antibodies. See this image? There is a blue color antigen on which there are various colored epitopes seen. So these are the epitopes, this is the antigen, and you see polyclonal antibody, which is a general response, it can bind to various epitopes. Whereas you see monoclonal antibody, it binds to a specific or single epitope. If you can understand this image, then your concept is clear. This is a quick differentiation between monoclonal and polyclonal antibodies. Monoclonal, because we are performing a hybridoma technique, right? So they are expensive to produce, whereas polyclonals are generally produced. They are cheap. We use them in vaccination programs. But monoclonal antibodies are expensive and we use them in cancer treatments and different diseases, which I will be discussing in the next part of discussion. As I already discussed, single antibody species, there are mixed population of antibodies you will get, will only bind to one specific site or epitope, and they may bind to different areas or target molecules. Even with a small change in the epitopic structure, these monoclonal bodies they're highly specific. They're intolerant of small changes. But polyclonal antibodies, even if there is a small change in protein structure also, they act. That is what happens with vaccination. Influenza vaccination, there is an antigenic drift and shift. So small change in antigenic structure, still the vaccine can act because vaccines are mainly made up of polyclonal antibodies. So there is an extensive list of monoclonal antibodies which are used in the modern medical practice. I think you should be aware of all these things because there are so many questions asked in this regard. But what I'm trying to do is I'll give you these lists. There are 3 slides, number 1, number 2 and number 3. So let us look into all these 3 slides. We'll be focusing on more on what is the target antigenic epitope. Okay, so based on that I'll give you what is the indication of the drug. But this second column you see here, the type of monoclonal antibodies needs a special mention. You may synthesize a monoclonal antibody directly from a human being, that is a humanized form, or we take some component of human and some component of another animal, we call it as a chimeric antibody. Okay, so when I say chimeric, it is a hybrid. When I say human, it is exclusively derived from a human source. So we'll follow the alphabetic order. We'll start with abciximab. So abciximab is a chimeric and mainly it inhibits GP2B3A. And you know what? This is important for platelet adhesion, aggregation and activation. So if you inhibit this, you can use it as an anticoagulant, right? So monoclonal antibody kind of anticoagulant is abciximab. Then is adalimumab. It is a humanized form. It inhibits TNF-alpha inhibitor. So most of the autoimmune disorders, you take example of rheumatoid arthritis, which is a classic thing. We inhibit TNF-alpha signals there. So this is adalimumab. There are many more TNF-alpha signals. Let me finish off certolizumab. This is also a TNF-alpha inhibitor. See this? And we use it exclusively in Crohn's disease. So we are done with 3. Let me go back to the alphabetic form. Alemtuzumab, very important. It is used in CLL, chronic lymphocytic leukemia, and the target is CD52. Next is basiliximab, IL-2 RA receptor, transplant rejection, not important. This is more important. They are asking in the exam. What is the mechanism of action of bevacizumab? It is a humanized form, and very important is it is vascular endothelial growth factor inhibitor. So it is used in cancers, colorectal cancer, and very important is ARMD, that is age-related macular degeneration, which is an ophthalmological condition. And what happens in age-related macular degeneration? There is vasculogenesis. The vascularity in the retina leads to damage because these newly formed vessels are highly permeable. They are leaky, leading to the retinal hemorrhages. So this is very important, bevacizumab. And cetuximab, commonly used for cancers like colorectal and head and neck. It is against epidermal growth factor, EGF. Last in this slide, daclizumab. It is also IL-2R alpha receptor antagonist, that is CD25. It is used in transplant rejection. The next slide, eculizumab, very important because it has a peculiar indication. It is used in paroxysmal nocturnal hemoglobinuria. It acts against complement system protein. Efalizumab, not important, latest. It is used in psoriasis and the site of action is CD11A. Zemtuzumab, it is used in AML, CD33. Ibritumumab, not important, it is used in NHL, but this is very important, infliximab. We come across this infliximab commonly, and we got almost, this is the third when it comes to TNF alpha signal. We use it in autoimmune disorders. And murubbimab is used in transplant rejection, not important. This is important, natalizumab. The many MCQs asked in this. Natalizumab is actually used in multiple sclerosis. And what is the site of action of natalizumab? Very important. It is alpha-4 integrin. Try to remember this. This is important. Alpha-4 integrin. We have come to the last slide. Just concentrate here. This is very important. Omalizumab, because we use it in asthma management. It is monoclonal antibody against IgE. IgE means allergic, right? So this is omalizumab. This is not important. Palizumab, panitumumab is also not important. This is important because it is used in macular degeneration again, because it is a vascular endothelium, like bevacizumab. Ranibizumab is also used in macular degeneration because it is a vascular endothelial growth factor inhibitor. Rituximab is commonly used in clinical practice for NHL, important, and focus here again. Trastuzumab, very important because we use it in breast cancer. ERB-B2 monoclonal antibody. So let me summarize the important things. What you need to remember from these. Try to remember bevacizumab, very important, and try to remember eculizumab because it has a specific indication, and infliximab, and very important natalizumab, and also very important is omalizumab, and the last one trastuzumab. They have some characteristic indications and questions are asked from these topics as MCQs. So this will conclude our discussion on monoclonal antibodies. What is the basic difference between them and polyclonal, and what are the major monoclonal antibodies in common practice with their indication. Hi doctors, let us discuss about interferons in this video. When I say interferon, the first thing that should come into your mind is they are antiviral. So they act against virus and they kill it. Let us discuss about the classification of interferons. What are the properties? What is the mechanism of action? And what are the clinical uses? And we'll conclude the session by discussing the adverse effects of interferons. As said earlier, interferons are mediators of early host defense against virus. So whenever a virus enters into the body, the immediate defense mechanism of the body is by production of interferons. And these interferons are classified into three types. One is alpha interferon, other is beta interferon, and the third variety is gamma interferon. Among these three, major antiviral properties are exhibited by these two, that is alpha and beta. Alpha means it is elaborated from the leukocyte, so we also call it as leukocyte interferon. Beta is elaborated by the fibroblast, we call it as fibroblast interferon. And gamma interferon is also known as immune interferon because it is produced by immune cells, that is T lymphocytes. When it comes to the structure, Alpha interferon is non-glycosylated protein. It is not a glycoprotein, whereas beta and gamma are glycoproteins. You can see this is glycoprotein and this is also a glycoprotein. So alpha and beta are antiviral as discussed earlier. Whereas gamma, what is the major mechanism of action? What is the major use of gamma? Gamma interferon has immunomodulatory properties. So immunomodulation and anti-proliferative function is more than the antiviral defense. That is the reason we use it in a peculiar disease called chronic granulomatous disease, where there is an issue with immunity. That is, there is defective phagocytosis. So now you are thorough with the different classes of interferons. Let us now discuss about the properties of interferons in general. Interferons produced by the body, these are non-toxic and non-antigenic. So they cannot stimulate production of antibodies. And so there is no antibodies, they directly act, these interferons. And these are species specific. That means human interferons can act only in humans. So the animal interferons cannot be injected into the human for antiviral property. After the production, after the reaction, they are inactivated by proteases in the body. So they have a short duration or short span of action. And when it comes to stability, they are stable at a wide range of pH, that is ranging from pH 2 to 10. So these interferons are highly stable and we cannot dialyze them. They are non-dialyzable and non-sedimentable. We cannot even centrifuge them and collect them. One peculiar feature about interferons, the last one is, they are endogenous pyrogens. Interleukin 1, IL-6, TNF-alpha and interferon, they are all endogenous pyrogens. They elevate body temperature. So these are the properties of interferons. Now let us look at the mechanism of action of interferon. Let me take you through this diagram. Here is the virus, the first step. Whenever virus enters into the host cell, it releases its DNA or RNA. Later, there is synthesis of new nucleoside along with the capsule and there is viral assembly, and after that there is release of virus into the surrounding environment and this new virus is taken up by another cell. This is how the virus spreads from one part to the other part. When this mechanism is going on, the virus also in turn activates the genes of interferon. So the virus acts and it activates on the genes of interferon, and whenever the IFN gene is activated inside the cell, it releases interferons, and these interferons, they bind to interferon receptors which are present on the adjoining cells. And when they bind to interferon receptors with the help of JAK-STAT pathway, what is that pathway? JAK-STAT pathway, they convey signal to the DNA of the next cell, and there is synthesis of new antiviral proteins, and these antiviral proteins, they destroy viral particles. So after infection with the virus, the spread of the virus to the next cell and its action is disturbed by production of these interferons, and the production of interferons is actually aided by the virus itself, because the virus is causing activation of the interferon genes, release of interferons. Later these interferons are acting on the interferon receptors, there is a signal going to the nucleus of the cell and there is release of antiviral proteins. We call these antiviral proteins as interferon induced proteins. Now let us look into the uses of interferons. What is the method of administration of interferon? Can we give it orally? No. Interferons are inactive orally. They are given either IM or IV, but the latest pegylated forms are for better action, longer action, better T-half. So these pegylated forms, even though they are costly, they are used nowadays, and these pegylated forms can be given subcutaneous, not daily, because pegylated forms have a longer duration of action. You can give them twice or thrice weekly in a subcutaneous form. So interferons are not active orally. So what are the different uses of interferons? You can see here, as I already told you, interferon alpha is the only form which is synthesized in a recombinant way. We do have interferon beta and gamma, but when I say interferon uses, we are primarily focused on interferon alpha, and what it acts on two important conditions. One is diseases caused by viruses and also malignancies. There are two important forms of alpha, interferon alpha 2A and alpha 2B, which are used in clinical practice. Alpha 2A is active against non-Hodgkin's lymphoma, hairy cell leukemia, CML, AIDS-related Kaposi sarcoma, and chronic hepatitis C. Whereas alpha 2B is active against follicular lymphoma, hairy cell leukemia, AIDS-related Kaposi sarcoma, malignant melanoma, condyloma acuminata, chronic hepatitis B, and chronic hepatitis C. So your take home is, just remember this: interferon alpha 2A and 2B, they are active against majorly hepatitis B, hepatitis C, and also AIDS virus, that is Kaposi sarcoma. And in the malignancies, try to remember NHL, follicular lymphoma, and hairy cell leukemia. Nowadays we are using, even in resistant cases who are not responding to podophyllin. Podophyllin is the drug of choice for condyloma acuminata, but in resistant cases we are giving interferon alpha 2B, and even interferon alpha N3 is also in common practice. Let us quickly wrap up the use of interferon beta 1A 1B. It is used in relapsing and remitting multiple sclerosis. Just remember, interferon beta is used in multiple sclerosis. And at the starting of the discussion, I told you that the predominant major use of interferon gamma, which is not antiviral, right? It has an immunomodulatory action. And this interferon gamma is used for management of chronic granulomatous disease for ages. Okay, so these are the uses of interferons. Now let us conclude the session by discussing the adverse effects caused by interferons. One thing very important which you need to remember is, it causes flu-like symptoms. Actually, flu is caused by majorly virus. Though it is antiviral, it causes flu-like symptoms. That is fatigue, body aches, pains, weakness, malaise, fever, dizziness, anorexia. All these are like flu-like symptoms. They develop within few hours after injection. But as we go on giving injections, slowly the adverse effects they wean off. Next important side effect is, interferons are known to cause neurotoxicity, myelosuppression, and they may cause thyroid dysfunction. So when I say thyroid dysfunction, it can be both hypo as well as hyper. So these are the important adverse effects associated with interferons. So in this video, we covered classification. What is the difference between alpha, beta, and gamma interferons? What are the properties? Main is they are species specific, and it is also an endogenous pyrogen. When it comes to mechanism of action, you have to remember this JAK-STAT pathway, very important. And uses are specific. Interferon alpha, we use it for cancers and viral infections. Beta is for multiple sclerosis, and gamma is for chronic granulomatous disease. And adverse effects, please remember, flu-like illness.